if you can see it here, it should be fine. This one, right?
Yeah, thank you very much.
Sorry for that.
Yeah.
Can we start?
It should be fine, yeah.
Okay. So, good afternoon. Just a quick introduction. I'm IMBRSea from the University of the Algarve, and we have here other members of the jury. Sara from the University of Oviedo, Fernando, yes, from the University of Oviedo, and Carlos Stemmer from the Sorbonne. So you have 15 minutes to give the presentation, then we'll have 15 minutes of discussion, and the members, people in the audience can ask questions, and people online, following online, can also submit questions on the chat. So please start.
Thank you very much. So my name is Georgina Vickery, and today I'll be talking to you about time series expansion, specifically how during my thesis I've successfully developed a new method to convert captures from trawl and trap gears to a video standard. I did this using the snow crab stock in the Barents Sea. I chose to use this stock because it's a very lucrative fishery, and in addition it can also be considered to be quite a politically contentious stock. So taken together, these two things mean that it's very important that we know both how much of it we have and also where it is. So to assess this stock, there's three main sources of data. However, it's actually very difficult to try and amalgamate all of these three time series and put them together. The reason for this is that they're all conducted at different times of the year and they're done using different gear types. So to try and elucidate this, the top two surveys there use something called a Campbell and bottom trawl, whereas the bottom survey, the snow crab survey, uses a different type of trawl called an IMBRSea trawl, which is very different. It also uses video data and snow crab pops. The use of video data is really exciting because it gives you 100% catchability. What this means is that as the video goes along the seabed, it captures every single crab, or you can count every single crab in its path, providing it's good visibility. This is different to a trawl when we don't actually know how many crabs we don't catch. So this picture here on the right is very blurred. It shows you the distribution of all of the stations together from the three surveys. These red dots, which are not visible, show the small area where we do actually have video data. And this already highlights the lack of spatial overlap between the three surveys. Partly due to this, the current approach to assessing this stock doesn't really use this really high quality video data. Instead, it mainly uses the Barents Sea ecosystem survey and does a quite crude index of catch per hawk. If we could actually combine the data from all three surveys, the stock assessment would be a lot more reliable because we just have more data points. But there's, I'm using the snow crab survey as an example, but there's a lot of issues with amalgamating time series data that are persistent across different types of surveys all over the world. And it's that sometimes we have a lot of holes in our time series, like maybe a vessel broke down, maybe there was a slight change in a gear. Sometimes in the Barents Sea we can't access all of the stations because of ice cover, so we just simply can't get them. So if we could compare data between different gear types and then actually amalgamate the time series together, we could fill in the gaps and end up with a much more reliable picture of whatever ecosystem we're actually looking at. So I'm going to focus now for the rest of the presentation on the snow crab survey because it included video data. So these are the three gear types that we used in the snow crab survey. If I wanted to go ahead and calculate a gear conversion factor, the first thing I need to do is consider effort. How much effort do we put into trying to catch these crabs? For a pop gear, typically we do soak days, so the number of days it was deployed for. For the trawls, we would use something called swept area, which is the area covered during the towing. But within these calculations, there are three assumptions which are very rarely actually explicitly considered, but people do this. The second one, catching efficiency of a gear, is possibly the hardest to take into account. Because, for example, we know for sure that when there's higher current velocities, our pots catch more crabs because the bait beam is spread further. But it's very difficult to actually quantify this. Furthermore, for example, when we use a trawl gear, we know it operates slightly differently on different sediment types and also at different depths. And again, this will differ with the length of each crab, the size of it, but again, we don't really know the extent of this. We can't put a number to it. So it's very difficult to actually take these assumptions into account. People have tried, obviously. So the snow crab survey did try and do this experimentally, and they put a video station as close as they could to a trawl or a trap station. this can result in, when this is done in other cases, it results in one single scaling factor that's applied uniformly to all of the data, regardless of the conditions, which obviously ignores the changing availability of the species to the gear. You can also apply a range of reductionist modelling approaches, where the modeller will pick a few environmental variables that they think are important before they start off, maybe sediment, maybe temperature. They might explain the difference in captures, and then they'll just use these. And again, they'll ignore anything else that they didn't think was important at the start, again resulting in quite a lot of error, and you're really reducing quite a complex system. So this leads me quite neatly onto the aim of my study, and it's to try and use machine learning to try and incorporate more of this environmental complexity into gear conversion factors. So the overall aim is to develop a reliable transferable method to scale the captures from trawl and trap gears to the video standard. I tested this using two hypotheses. Specifically, does the incorporation of environmental data actually improve the accuracy of my model, of my conversion factor? And secondly, once I've converted my data and I've expanded my data set into a higher quality amalgamated data set, does it actually improve the overall predictions of snow crab abundance? Like, so do we actually need to do this at all? Do we need this extra data? So to give you an overview of my methodology, I started off by developing 3 baselines so that I could evaluate my hypotheses. Secondly, I developed a conversion factor, which incorporated this environmental complexity. And lastly, I did a final model where I used my video data and my converted trawl and trap data to predict across the Barents Sea. So to go into a bit more detail on my first approach, I used the Haversine distance formula to calculate the distance between each video station and the nearest trawl or trap station. And I then did a linear regression between these. So these are the results. On the right here we have the trawl data, and you can see it's got a very, very low R squared. The trap data here, this is for 5 nautical mile distance by the way, has a higher R squared of 0.6, but 24 data points, so not particularly reliable. The results for the 3 nautical mile distances were the same, but just with even fewer data points. To move on to my second baseline that I tried to calculate for the conversion factor, I developed something called a random forest model. And I used this model to use the video data, which is known to be true, in theory, to predict the truth at each trawl and trap station. The aim of this was to try and check if in the previous example you just saw, if the low accuracy was due to the distance between the stations, or if this low accuracy was caused by perhaps environmental conditions. So I'm going to explain briefly what the random forest kind of looks like. This is one single decision tree, and the forest is a group of these trees. And a random forest is a supervised technique. This means it will never predict outside the range of the data that I give it. I chose this model because it's used very widely in the literature, and it tends to outperform its similar machine learning counterparts, and also GLMs and GAMs were tested on similar data sets. For the predictor variables, I chose to use the mean monthly values of my environmental predictors because these are less susceptible to daily fluctuations. I also tested for the significance of interactions between these, and I tested for these using both spatial and non-spatial evaluation metrics. This just shows the breadth of the environmental data that I tested for in each model. For the temporal variation, I used the Julian day and calendar year of each station, and for the latitude and longitude, I took the lat and long of each station. The model fit was improved by log+1 transforming my data, and I also very, very thoroughly tuned it using both manually and by using a grid search algorithm within R. And again, I use spatial and non-spatial cross-validation metrics because there's pros and cons of each, so by doing both, I more thoroughly evaluated my model. So this is a table that's going to fill up as I add models to it. And I've chosen to just show you the R squared of my first model because it's the simplest metric to understand, but obviously I did use more evaluation metrics than just R squared. So this result of 0.7 shows that overall I had quite good accuracy of my first set of predictions at each trawl and trap station. To try and compare this to the previous approach using the closest stations, we get this. So you've already seen these graphs here. And on the top, we see overall there was an increase in R squared when I used the random forest approach for the linear regression. For the trapped gears on the bottom here, we actually see a slight drop in R squared to 0.4, but we have a lot more data points. We have 100 data points. And this really shows that the data set overall is more robust. To try and add these into the table you've previously seen, we see with the closest stations approach, combining the gears, we have a very low R squared. When we remove the impact of distance between stations and increase the number of data points, I have actually increased it quite significantly because I managed to capture more variation in my data. For my third and final baseline, I used the same random forest model, M1, on a prediction grid across the whole of the Barents Sea to see how my abundances were. And this is what we get. Overall, there's more in the north than the south, which is consistent with the literature, and I have a maximum prediction of 250,000 individuals. This seems like a really big number, but in my raw data, I actually had values of 700,000, which is much, much higher. But however, the median value of my data set, my raw data, was 8,000. So this does capture the majority of the range of my data that I put in, but it's not getting the extremes. So to try and test my first hypothesis, which was, to remind you, does environmental predictors improve my conversion factor? I did this using a random forest model the same way that I developed the same way as I previously described for the first one. And here we go. So this M2 is my second random forest model and have an accuracy of nearly 100%, which is the reason for the drop here is because this is the testing data from the model. And this is very, very high, and I was very pleased with this. And the same pattern was shown with both the spatial and non-spatial cross-pollination. And this really, really emphasises to me that it's so important to include the environmental interaction with the gear and how that determines the final catching efficiency. To try and test my second hypothesis, I predicted again across the Barents Sea and made a second set of maps. So again, we had the same overall predictions per year, and again more in the north and the south. But you can see before there's quite a hard line at 28 degrees, and that's gone with more data points. And furthermore, I have a maximum prediction of 500,000, which is much, much higher. It's still not 700,000, but I actually only had about 5 stations that were between 500,000 and 700,000. So this really is getting the majority of my data. Again, though, it's not predicting zeros. The lowest prediction is 5 individuals, which again is something that needs to be considered. A final table that I'm going to show you, we have a final accuracy to evaluate this model of maybe 0.9. So again, that's much, much higher than my first attempt at conversion model with random forest, 0.7. And again, it's much higher than the linear regression approaches. So just to pull out some of the variables that were the most important in my model, the top 6 predictors were actually all geospatial variables. B temp here stands for bottom temperature, and that was also very important. Interestingly, sediment was the least important, which I was quite surprised by. But the reason for the importance of the geospatial predictors is because the stock is really patchy. So there was quite a lot of groups of juveniles, which also accounts for the really high values that I had. And it could also be that there was another predictor variable that correlates more strongly with my data than the ones I included. Temperature, we knew it was going to be important before we started, and I'm glad that it did come out to continue to be important. But it's really important to also understand that snow crabs show an overall changing preference for habitats during their life cycle. As they develop, they have a preference to move offshore into deeper waters with more stable temperatures that are slightly warmer. So when you interpret the data, you also need to consider the changing biological preference of the snow crab. Sediment actually agrees with the literature, the absence of this from top predictors, because the large body size of these megabenthos species means that it's not really constrained by a grain size. It's more driven by things like prey availability, because it can tolerate a wide range of environmental conditions. And so therefore this suggests that other variables with more biological basis should have been included, but it was very difficult to find these predictors on a small spatial scale on the time period of my model. And just to go over some caveats for the model, it's really important to note that it's the modeller's decision on what resolution to use of all of the variables. And this can kind of result in data dredging, where you find correlations where there are in fact none. And so you must be very careful not to come up with things just because you think they should be there. Also, the snow crab survey data is slightly biased in that it takes place in the main area where there's the most snow crab. So again, we have a few, we often get large clumps of the juveniles in possibly unsuitable habitat, which is one of the reasons why the model didn't necessarily pull out any one single predictor to predict the overall distribution. But again, this can be improved by the inclusion of length or maturity into the model. Finally, I'm currently developing a second model, which is a binary classification model to add into an ensemble, whereby I'll be able to finally predict the zeros and have more accurate predictions overall. So to conclude, both hypotheses were fully supported throughout the model, and I really want to emphasise that the inclusion of environmental data is a really big step towards being able to really amalgamate multiple time series. But we have to be careful to avoid data dredging and finding spurious correlations where there are none. And also, it's very important, if possible, to use an ensemble approach so you have the pros and cons of multiple model types throughout. Thank you very much for listening.
Thank you very much. You were just a few seconds over the time. So now I'd like to invite the members of the jury to, anybody has any questions?
Yes, sir?
Yes.
Okay, well, thank you very much, Georgina. This has been a fascinating trip to Northern Europe and that wonderful ecosystem. Wonderfully productive also, and some of the most important fisheries in Europe. So it's always nice to see that, somehow, the responsible and young people continuously pioneering and improving with new methods. In this particular case with machine learning approaches that, I don't know, right now we are experiencing some kind of verge or rise of comeback, at least for me, you know what I mean? I see there Leo Brigham paper from, in 2001, I was thinking, oh man, random forest again. That's like, for me it's more like a comeback than a revolution, but it's a very complicated technique, so congratulations for handling it so wisely and for this nice presentation. Having said that, I have two, I don't know, evil questions, okay? The first one, at some point, I don't know if I missed the detail, but you were doing some kind of key-fold cross-validation or out-of-back prediction or something like that, if you can comment.
Did you say forward-back prediction? What? Did you say forward-back prediction?
No, fold, key-fold.
Oh, no, I didn't use K-fold spatial cross-validation.
No,
I used...
Out-of-back.
Pardon?
Out-of-back.
No.
No?
I used the inbuilt evaluation functions within the spatial RF package in R. And what this does for the spatial cross-validation, it will split, I chose 29 independent spatial folds. So I split the data up into, for this I used the clustering, into 29 independent spatial clusters. So the 25% of the data was separated for testing, and then I used the remaining 75% to train the model on. So then when I tested it on this 25%, that 25% of data was spatially independent from the rest of it, because when you do non-spatial evaluation on spatial data, just by chance, if you plotted all those points, the training and the testing data that you used might be next to each other, which means the models almost had a sneak peek at the testing data, and so you get an inflated value. Does that make sense?
Yeah, makes a lot of sense. Okay, I think I will skip my second question to Jan, just to minor detail, or a very important question. If a manager asks you whether you should stop doing the traditional sampling protocol and incorporate the kind of approaches based on video or something, that kind of stuff, what would you tell him, and would you be able to put a number in terms of the money that they will save?
The money they would save? Well, I would say that if you, with the snow crab survey, they only count snow crab. So they have two observers and they sit there and they click away as they count, and they count it in real time, and they just count the snow crab. But if you could automate it and have some form of, well, it's currently being done, a deep learning way of calculating both also the length of the species as you go, you could do this for every species. And so the initial investment would be large, but the payoff would be huge because you would have really accurate data for a lot of benthic species. And video is being increasingly used also because it's a non-invasive technique. So, and there's obviously an increasing move to try and have less trawling, especially with scientific data when you don't necessarily need to be killing 5 tons of organisms in one go. So if a manager asked me if they should use video, I would say absolutely, and the more the better. Because the more research we put in to try and automate it, the more efficient it is, and the more we can do it. So it's a positive cycle, I think, for that. Does that answer your question?
Yeah, perfect. Thank you very much.
Okay.
Thank you for a very nice presentation. I would like to go back to the, let's say, the life cycle of the crab, but that's something you did not explain us.
I actually have this additional slide on that, I think. As in the changing preferences of it, or
Yes. And also the, the long-term dynamic of the population of the crabs.
So this stock is actually very interesting because it's not an invasive species, but it's been naturally expanding its range from its native area in around Canada and Alaska. So it's been walking over the top of Russia by adult migration, which is partly why this model is quite difficult because the snow crab sort of started here and it's been spreading across. And so as the adults mature, when the larvae settle, they settle in very cold water, and they prefer to have fine sediment that they can bury in. But this is also the preferred habitat of the adults. So as the juveniles develop, they are often outcompeted by the adults for this preferred habitat, which is partly why you then get groupings of juveniles on the less than optimum habitat because they're pushed out by the adults. Generally, for their food preferences, they eat anything, anything going there, eat it.
And anything which is alive, so it means alive prey, such as mollusk, or does it feed on the production of the surface layer because it's very productive there?
It doesn't directly feed on small items of detritus, simply because it's just too small. But anything chunky, it will get its teeth into. Each other, their own legs, they'll eat it.
Okay.
Very much. But they also show in both their expanded range here and their native range, they do move into deeper waters offshore as they get older. But they also show seasonal migrations inshore as well. And some of the really large adults, the adult males, they can migrate 170 kilometres inshore and out again.
Because the prey distribution was not in your model.
No, it wasn't
But
because...
it doesn't mean because there is enough food everywhere?
Well, in the Barents Sea, they actually overall, the average body size is larger than the average body size in its native ranges, partly due to more food availability, and it's not limited by food. But that being said, the Hardy 2011 paper I cited quite a few times, they found that chlorophyll was a top predictor of their model. They used chlorophyll because they couldn't get any kind of benefit productivity predictor. And therefore that kind of leads back to your previous point that perhaps the surface production would impact the distribution. I didn't test this, but it's on my to-do list, the model I'm currently redeveloping.
So now you are having let's say some indices of the viability, spatial viability of the food in the new version?
That's what I'm trying to do, but it's chlorophyll again, like you can look at it in the top layer, but by the time it gets to 200 meters depth, it's not in the same place anymore, and it's all been moved and changed and recycled. So I was also looking at the 3D habitat structure and trying getting some kind of biogenic structure, but again, that's quite hard to get on a small spatial scale. So
yeah, because I was surprised as you by the fact that the sediments were not really important, but the sediments are important for the prey of the crab.
But I think it's because they're so generic, they don't feed on one specific species, and they move around a lot.
Okay. Just a very short question on your graph. I did not understand what was units.
Oh, sorry, it was individuals per squared nautical mile.
Okay, but then you are also on the slide 9, for example. Yeah, what is a video CPU? Next one.
Video catch per unit effort.
But what's the because there is a big difference between 2,000 and 200,000. You know, on the Y and the X axis.
It's mostly because the traps have a very low catch rate angle.
The by factor of 1,000.
Yeah, and also because the videos were deployed. The videos capture a lot of the aggregation of the small juveniles, and these aren't picked up by the traps because the crabs have to walk through the trap and get in. And that basically means you have to be big enough to get in the trap to start with, but not too big, obviously. And the small ones, they just simply can't walk as far.
A factor of 1,000 is huge.
Yeah.
But can you really compare it as a 1-1 or not?
Well, this shows that no, you can't.
No, you can't, because you divide by something to get the value of the shot.
But also, a lot of my data from the video is really skewed by the the aggregations of the juveniles that were really patchy. I haven't got it here, but I made a histogram obviously of my raw data, and there's like 10% of my catches are 0, and the majority is kind of here, in like just like less than 8,000, and then I have the rest is a really long tail going up to 800,000 and 700,000. Which is partly why I have a weird distribution, which is why I want to also include the commercial data and the other 2 surveys, which is what I'm currently doing to try and sort this out.
Thank you. We don't have very much time.
Okay, yes, good question. Thank you very much for your presentation. And how did you choose the environmental variables? Were they all at the beginning or then training?
Well, I started off just using sediment and temperature from the literature, that's what everybody uses. But I very quickly realised there was a lot more going on, which is when I went in search of current velocity and ice cover. And then I just downloaded everything that was available, to be honest with you, so that I wasn't putting my opinions on the data. I wanted to be as unbiased as possible. But then I went through and selected for the ones that were significant in the model. So not every model had every parameter in it. So that's how I ended up with the variables that I showed.
Okay. And just out of curiosity, you said they are not using the video data.
Yeah.
Why they prefer the one?
They prefer the video data has they have a very small index of the video data that they do try and use, but the snow crab survey starts in 2018, but the best survey starts in 2004. So it's a much longer time series. So it's the assessment models picks up more on the longer time series, and the various ecosystem survey has a much larger coverage. Spatially, it goes much further north. So it's overall you could say it's more reliable. But I think it varies a lot, personally. So, yeah.
Thank you very much.
Okay, unfortunately we have to stick to the time, so the next person.
Thank you very much.
So the next presentation is by Karina, and so please you can start very on.
Good afternoon, everyone. My name is Karina Orsolini, and I'm going to present to you my thesis project entitled "Is a Basic Monitoring Protocol Effective to Provide Quality Information on Bent Indicator Species in Sidon Cays in Brazil?" So human-induced pressures, such as climate change, coastal development, overfishing, and nutrient runoff are affecting species abundance, distribution, and survival rate worldwide. This is even worse when local and global disturbances are acting together. To overcome such threats, the creation of marine protected areas become the typical conservation efforts to protect marine ecosystems against the cumulative impacts of human activities, with long-term monitoring programs allowing us to evaluate the performance of marine reserves, to assess changes in species abundance and the natural variability of marine populations, and to improve the management and conservation strategies. It is in that sense that the Monitora program was created in Brazil to monitor the biodiversity at the national scale, including the marine biodiversity. This program has a subcomponent for the reef ecosystems that are planned to be monitored by for a benthic protocol. And this protocol is being constructed by many scientists, including me, and many MP managers alongside Brazil. But for this construction, we need to take into account the premises of the program, the cost-benefits premises, such as the protocol needs to be basic, to be applied nationally, and to be of a long run. The targets need to be just a few to don't impact the budget and the logistics, and of easy identification to overcome the level of experience for the managers who will be applying this protocol. And finally, the criteria for target selection was based only in the species under fishing pressure. So with this work, we aimed to assess the information on benthic assemblages that could be provided by a basic protocol, such as Monitora, and to understand whether its results can produce quality information for the MPA managers in Brazil. But as I told you before, this protocol has not been implemented yet. So in order to understand if this would provide a quality information for the managers, we simulated the future results that this protocol would achieve, using the dataset of the MAARE project, which is a completely independent project that runs inside the Arvoredo Marine Biological Reserve since 2015 to today, but they have some missing information on the years 2017, 18, and 2021. The MAARE project protocol has a similar amount of epibenthic targets, but they have much more megabenthic groups, indicator groups, when compared to the Monitora. And this is the overall picture of the comparison that we conducted in this work. So for the epibenthic targets, we contrasted the 12 groups, indicator groups from the MAARE with the 8 groups for Monitora. And then for the megabenthic targets, we compared the MAARE with the 20 species with the Monitora with only 3 species groups. But since this reduction of targets was very drastic, we decided to create an intermediate scenario that we call it simplified MAARE, in which most part of the species of the MAARE were pooled into groups. And then we compared it 2 by 2. All the analysis was performed using our software. We tested for normality and homoscedasticity, but assumptions were not followed, so we used non-parametric tests. Descriptive analysis were done using box plots, and then we tested for variance in abundance across the years and then between years. And then the multivariate analysis, we used the PCOA, and we tested, then the statistical test was done with one-way and pairwise per ANOVA, and later on with SIMPER analysis. But unfortunately, I will not have time to present this part of the results, but if anyone wants to know more later, I'm glad to answer the questions. So I divided the results and discussions into 2 parts. The first one regarding the epibenthic results, and then I'm going to move on with the results of the megabenthic targets. So let's start with the epibenthic targets. These five groups are common targets for both protocols, MAARE and Monitora, and so the Monitora protocol here would achieve similar results as the MAARE, and in which more than 80% of the substrate coverage is dominated by algae, especially turf and by zooantheres. And even if we can detect significant decrease and increase in abundance for some of these groups, we cannot have an inference on the fluctuation pattern because of these missing years when the serving could not happen. And we understand that we would need a longer temporal series to understand this variability in abundance for these common target groups. But the strong point of Monitora in this case is that they are planning to survey the algae communities separately. Aude et al. Proved that most part of the Brazilian province is dominated by turf. And so having a protocol, monitoring protocol that are serving the algae communities separately would allow a further comparison for the benthos community that could be applied locally, nationally, or even globally. And would also allow us to understand temporal shifts of these communities if it happens. On the other hand, Monitora is not including groups such as ascidians and sponges, filter feeders. And when they do this, they lose the capability of tracking water quality. Wilkinson and Cheshire suggested that with the increase of nutrient runoff, there is a concomitant increase in sponges abundance and biomass. And then Lambert suggested the same for ascidians abundance. So if we have monitoring protocols that are tracking this, the abundance of these species, we would have the information we could try to correlate with the water conditions in the study sites. And this is even more important when we know that the Arvoredo Marine Biological Reserve and many other MPAs in Brazil are located close to the shore, and then they are susceptible to runoff and coastal pollution. So that's why we, in our project, we suggested the inclusion of filter feeders category. And even if this alert was needed for the epibentic targets, we understand that the monitora would achieve or highlight the main changes on the groups that are structuring the landscapes in Brazil, the algae communities. So we found that the monitora would be a suitable basic protocol to monitor the epibentic targets. Now let's move on to the second part of the results regarding the megabentic targets. Just refreshing the memory, MAARI is the protocol, the very complete one with 20 species, and then it allows us to assess the abundance at the species level. So for example, with the MAARI protocol, we could see that there is a tendency to increase in abundance of the exotic bryozoa, the Schizophorella elevata, and then we can also detect the appearance of exotic, invasive, and tropical sea urchins in the study site. Here we can see with the outliers that the exotic Ophiuroid, Ophiodella mirabilis, and then the invasive sun coral Tubastra coccinea are appearing in our transects from 2022, and there is a tendency to increase of the sun coral in our study site. Moreover, we have also the appearance of species of tropical sea urchin species, Pseudobolete maculata, and species of Giardamatidi family, appearing in our transects from this year. So even if we have a very detailed information, what are the species that are appearing in our transects or increasing in abundance, this turns to be a constraint for the monitora program, given to the premises that I said to you before. And then with so many target species, that could impact the logistics in the field. But on the other hand, when we take a look on the results that Monitora would achieve in the future, they are planning to survey only blacky urchins, the sea cucumber Olotura grisia, and leader. And when they do that, they have only information on 2 biological groups. And we know that they are going to lose information on the ecological disturbances that are happening in the study site that I just showed you by the results of the other protocol. So because of that, we understand that Monitora would provide just a limited information for the managers. Moreover, they are planning to survey exotic and invasive species using the presence and absence method. But we already know from the study of Castro and Pires that the genus Tubastria invaded Brazil in the late 80s. And then Lindner proved that the species Tubastria coccinea, the sun coral, is increasing in abundance in the south of Brazil. So we already know that there is this invasion happening in the study site, and we need to have the traceability of these species, how they are spreading, how they are increasing in abundance. So that's why we suggested Monitora to change the estimation of exotic and invasive species to an abundance estimation. And finally, they also don't include the non-indigenous species, such as the sea tropical ones. But we already know from the study of Andrew and Byrne that the species of Giardamatidi family that moved from Australia to Tasmania led to a catastrophic ecological grazing impact on the algae communities, and this could happen in Brazil with the arrival of these species. And then we also know that there is the species Pseudobolete maculata was first recorded in the southern West Atlantic in Brazil, in São Pedro and São Paulo archipelago, and right after in the states of Santa Catarina and Rio de Janeiro. So these are showing us that there are signs of tropicalization happening in Brazil. And this was proved by Silva et al. In this year that the tropicalization process is happening in the study site in the Arvoredo Marine Biological Reserve. So it is of paramount importance to understand the ecological implications on ecosystems functioning with the arrival of such species in the temperate areas of Brazil. And that's why we suggested the inclusion of tropical species to improve the information that would be provided by the protocol. So with all this information, we understand that Monitora would lose valuable information if they apply this just basic protocol. And then let's take a look in the last protocol, again, refreshing the memory, simplified MARI, we pulled most part of the species into groups, and then even if we don't have an assessment, an abundance assessment at the species level, when we put the species in groups, the statistical analysis detected variance in abundance for some of these groups, including the group of the exotic invasive species and the group of the tropical urchins. And this could provide a red flag for the managers to trigger a further investigation on what's going on in the study site. What are the species that are increasing in abundance and what are the species that are appearing in the protected areas? And furthermore, we understand that if resources are being applied to go in the field and to make a monitoring protocol, there is no extra costs for the managers to just sample a bit more groups that could provide them much more robust data. So that's why we understand with these results that simplified MARI would achieve very similar results as the MARI, the more complete one, and they would allow to highlight the main processes happening in the study site. And we understood that among the three protocols, the simplified MARI would be the most suitable to monitor the megabenthic targets. So concluding, we understood that the Monitora protocol would be a suitable one for monitoring the epibenthic targets because they provide information on the communities that are structuring the landscapes in Brazil. But on the other hand, it seems to not be true for the megabenthic targets, and to improve the quality information that they would provide by their protocol, we suggested to increase the targets, including non-indigenous species such as the sea tropical ones, and to also change the assessment of exotic and invasive species from absence and presence to an abundance assessment. I appreciate your time. Thank you so much.
Thank you. That was well within the time. Questions? Who would like to start?
Just can start again, but no sense.
I can talk.
Okay.
So thank you very much for this presentation of a new continent. We have been out of Europe, so now we are in Brazil. I would say that in terms of format, I like your slides, only the color, but I don't know if it's due to the...
Yes, the projector.
It's a projector.
Yeah, unfortunately the picture is very nice here, but you cannot see it.
Okay. What I was lacking a lot is some more information about the sampling, because you always show 4 sites, but we don't know where they are, we don't know their context, we don't know that, which is very important, because then we ask ourselves what is the quality control of the data, and how can we be sure that 10 here can be compared to 10 here, you know? So, yes, please, if you can explain more about this.
So the MAR project, they sample 4 sites within 3 localities, also always in the same depth strata from 1 to 8, and for each site we have 3 transects happening. And the methodology for serving the both targets, they slightly change. For the epibenthic targets, the diver is recording the type of substrate that is exactly under the metric tape every half meters alongside the 20 meters length transect. And for the megabenthic targets, all the target species were counting along the 20-meter length transect and counting 1 meter with each side, totaling 40 square meters.
And what are the units of the graph when you show us time series? What are the units?
The time?
When you show us the time series, what are the units?
The number, we have 12 transects for...
But maybe you can go to the, show me a slide with the time series, the number of individuals.
A time series?
Yeah,
like that?
What is the unit of black?
It's abundance. It's just the number of individuals in 40. Yeah, in the 40 meters, the square meters.
Okay.
Yeah.
So there is no square meter and.
Yeah, I should present that in the units.
It's
It
number
is-
of envelope per square meter?
Yeah, no,
So
for
it's
40 square meters. Yeah, yeah, for each dress.
Okay, and how many times you do it per year? Is it always the same season?
Yeah,
Is
it's
there
always.
any change due to the seasonal effect?
No, there's no seasonal effect. They always serve in the summer, and usually in the first 2 months they complete all the sites, so you don't have the seasonal impact.
Okay. Okay, it's the same. It's finished for me.
Okay.
Thank you. I really like your presentation, thank you. And I only have a question regarding the non-indigenous species, because you are talking about one that is already established, right? With
The invasion?
yeah.
Yeah. You mean which one? The tropical urchins?
Yeah,
Mm-hmm.
the one that entered in the 2000.
In this year, actually, yeah, we don't have much information because this was observed this year.
Oh, okay.
Yeah, and then we were very concerned about the situation because we are doing the surveying with the managers of the MPAs. And then when we saw them, we got very surprised. So that's why it came from there, the idea of this project, to try to understand what kind of information the basic protocol would achieve, because they are not including these such species.
So maybe they could include something like a list with species that are already established, so you can quantify for abundance and distribution and everything?
Would be the ideal, yeah, but then they have the premise that I told you, the target selection, they need to be only the ones under fishing pressure. So since this species is not an indigenous species, is not, at least in the study site, has not been recorded, we don't know yet how this is going to be. They probably are not under fishing pressure, and this needs to be followed by the program, you know?
Okay,
Yeah,
thank you.
unfortunately. That's the point. We wanted, maybe with these results, show the implications on not including such a species, you know? Mm-hmm.
Hopefully.
Yeah.
Okay.
Well, thank you, Karina. Thank you for the beautiful presentation. I had the feeling I need to apologize, as a local, for the quality of the projections, considering that you are talking about perhaps some of the most specious ecosystems in the world. That's how I reason that you just need to focus on a few species, because how many species do you have there?
It depends for each target. We have for the Monitora or
No,
the...
I mean, I understand that Monitora focus on just a subset of the community.
Yeah.
But, if you try to sample the...
Oh, it's too much. We would not have time. This would impact the logistics in the field, and I know this by like
An
my...
idea about
Of in species, how many species? Ooh.
benthic, macrobenthic?
I know that they have, in the marine reserve, they have more than 1,000 species, but not only benthic ones,
In terms of bits of information,
In terms of
bits of information.
bits, what are bits of information?
In terms of, don't worry. So we are talking of something similar to a tropical forest or a coral reef.
Not exactly. These are rock reefs, rock systems.
Okay.
If we survey this in the northeast of Brazil, we would be talking about rock
In terms
reef
of
systems.
the amount of species, the amount of...
They would have much more in the northeast, but
Okay, so now you're ready for that question. So the point is, you just focus on a subset of the community.
yeah.
That will allow you to realize, for instance, that some species are disappearing or the diversity is going down.
Yeah, let's try to get the most important ones that would provide this information.
Yeah, but that means that some of them are not important. And, that worries me. That's really,
Sorry?
that's concerning. That means that some of them are not important.
Yeah, all of them are important, but we need to choose some of them because we have time, we have resources, we have a lot of constraints for going in the field and trying to survey all of them. Like, for example, I did the black urchin surveying and I barely had the air in the tank anymore because it was so many that if you count all of them, you just left some behind. So you need to be careful on what are the species that you were choosing for your monitoring. And I'm not the person that are choosing them, I'm just, yeah.
If you have, I don't know, more resources, what would you try to incorporate first to your sampling program?
For the sampling protocol,
or for the monitoring program?
I would try to add another session for the samplings.
Not another
I don't
technology.
know if I would, what?
Not another technology.
Yeah, we could try also to go for another technologies, try to survey more. Yeah, like maybe using ROV or other type of technologies that would allow us to get all the information, not all, but at least the information from the benthic organisms that are over the on the substrate, something like that.
Thank you very much.
Yeah.
Congratulations again.
What about other taxonomic, like fish, for example? Is there another program for the?
Yeah, they are including. So the reef ecosystems, they are targeting fish and invertebrates, the benthic ones. But I could perform a similar work using the fish as well. But this data set from the fish belongs to another researcher, to another lab, and then I will need to try to bring him to my project. And then, to be honest, I'm not familiarized with the fish.
Well, I'm just curious to know if they also use just a subset of species, because to me that would be very strange.
Yeah, they are also trying to select, the protocol is under construction now. So we have many scientists that work with fish as well, trying to find what are the most important fish, the ones that could bring more insightful information for the communities. Yeah, so they have also the fish assemblages serving for this project.
Any questions from the audience?
Yeah, but maybe for the audience.
I think there's a question. Yeah.
Cool.
Who is paying for and managing these programs? Is it the Brazilian government?
Yeah. So there.
The federal government or the local?
It's the federal government. There is it is a Brazilian autarky that belongs to the government that are responsible for managing all the conservation units in Brazil, including the NPAs. And then before the MARI project was financed by a private company that was exploiting oil close by the marine reserve, and that was obliged to pay for a study to understand what would be the impact. But then they finished the project. That's why we have 2 years that are missing, because the project ended, and then there was another negotiation that was done with the managers of the marine reserve. And then this Brazilian autarky kept the monitoring to them. And then it kept running, and then 2021 was due to COVID. Yeah.
Thank you.
Here.
Well, yeah, my question is related to what you didn't show us, but proposed to show us, which is the multi-parametric analysis that you did.
The PCOA.
To what, so maybe you can show us if you did explain something more about the relationship between, those different groups, because here we see them individually, but when they are put together in a community, did you learn something more?
Yeah.
Did you learn something about the environment or not?
No, not about the environment, but we found that.
Maybe you can show us.
Yeah, I can. For example, this is the PCOA of the epigenetic targets. So the pattern of the results were very similar from what I show you in the box plots. So the epigenetic target didn't have much information lost because the number of targets were very similar. Okay, so you can see, I don't know if you can see properly because of the light, but you can barely see that they changed it. The only thing that really changed was like the space between them. It means that for each polygon is 1 year. Okay, so this is very clear that we have the assemblages of benthic communities. They change over the years, between some years actually. That was detected by the Permanov analysis, and this is clear here when we have completely separate polygons. This was the most, the highest result, the highest similarities found in our groups.
And what was the factor in those changes?
The groups that were leading, it was mainly the reduction of macroalgae. But
due to what?
Yeah, so I didn't investigate the reasons why the communities changed, because I would need to go in ecology and understand the interactions among this.
Which is interesting.
It's super interesting, but you know, we had limited time to do. And I would love to do next, maybe, or even a further step.
Environmental data are gathered.
Sorry?
Environmental data are gathered at the same frequency as this, let's say, community composition. Do you have a temperature? Do you have
Yeah, they have logs to temperature, they have many other oceanographic parameters that they collect, but this is really just a subset of a huge data set because the aim was really understand what are the information provided by the monitor, you know. Yeah, but the pattern was very similar. Even for the megabentic targets, we saw that with the drastic reduction of targets, the PCOA also changed significantly. And then even the polygons were more overlapping, so the communities turns to be more similar.
Okay, there's no questions on the chat, so we're almost out of time. So
Okay, thank you so much.
Wait 1 minute.
Yeah.
Okay, so our final speaker for this session is Sarah Elke. So, Sarah, you have 15 minutes.
Okay, so hi everyone, I'm Sarah, and I'm going to talk about the effects of patron-client relationships on climate change responses of small-scale fisheries in Nayarit in Mexico, and I hope you enjoy. So, small-scale fisheries, this is the definition by FAO, so I want to highlight that traditional fishing, mainly by families, relatively small amounts of energy, and mainly for local consumption. So, why are small-scale fisheries important? Well, they're incredibly important for coastal development, economic security, and sustenance worldwide, and specifically within Mexico, they make up 97% of the marine fleet. So, they're incredibly important. And to add to that, where Nayarit is on the Pacific coast makes up 89% of the total catch volume yearly. So, they contribute incredibly to the Mexican fisheries. Despite how important they are, they're also really vulnerable to the effects of climate change. And specifically, literature at the moment is highlighting that there's a lot of exits from the tropics in terms of fishery species, but there's not a lot of entries. And the reason why we pick Nayarit is because it's a hotspot for climate change, which means it's warming faster than 90% of the rest of coastal places across the world. So what is happening here, we can kind of see it's maybe a prediction as to what might be happening in the future in other small-scale fisheries. And a little bit about the history of governance in Mexico and the social organization that is there. So from the 1930s, there was cooperatives, and they had the monopoly over permits. So you had to be in a cooperative to obtain access to fishing permits. And this happened from 1930s until the 1990s, when there was a reform in policy. And then the permits became competitive. So anyone had access to them that could apply. It wasn't just for cooperatives. This also kind of created a problem because there is a severe lack of permits. They're really difficult to obtain for fishers. There's not enough of them and not enough people have them. And because of this issue, the patrons kind of came about. So this is another type of social organization. And patrons are people that have access to permits and they can provide fishers with access to permits in exchange for other benefits. So fishers can provide loyal workforce and political backing in exchange for permits, gear and boats and stuff like that. However, we hypothesize that because of this necessity for permits, there's potentially a power imbalance between the patrons and the clients, the fishers. So, in addition to this sort of power imbalance, there's another factor that can affect how they adapt to climate change and how they're responding, and that's adaptive capacity. So this is the definition, and it's basically just a fisher's ability to respond to the changes that they're perceiving in the environment. And Sinner and Barnes, they created 6 domains that make up adaptive capacity. So, for example, flexibility. Small-scale fisheries are incredibly flexible. This is because they change gear a lot of the time, they change species, so they're highly flexible. However, because of this, they're also highly vulnerable because of the changes that are going on due to climate. That's also a vulnerability that they face. So, if we combine these two things together, the social organization changes that have been happening in Mexico, as well as the adaptive capacity, I have my 3 research questions. So the first one is, what type of social organization exists in Nayarit, and what benefits are exchanged between the patrons and the fishers? And then the second one is, do fishers in patron-client relationships in Nayarit differ in their adaptive capacity to other types of social organizations, so fishers in cooperatives or independent fishers? And then lastly, are fishers in these relationships responding differently to climate change than other fishers in different types of social organization? So, in September to November in 2021, there was 445 interviews across 6 villages in Nayarit, here on the western coast of Mexico. And the fishers were divided into 3 types of social organization, and these 3 types have been seen in previous literature before when looking at this type of thing. So we have independent fishers, those working within cooperatives, and those within patron-client relationships. And across these 3 groups, we hypothesized a scale of autonomy. So we think that fishers in patron-client relationships are likely to have less autonomy than fishers that work independently or those within cooperatives. So that's the first part of the first question. And to answer the second part, we have these 8 benefits that they exchanged. So they are definite categories also used in previous literature. And they may seem quite distinct, but I want to mention that they're highly correlated. So for example, if fishers borrow money from a patron, they're obligated to sell their catch back to that patron. And bait, petrol, and ice, they're regularly used all together. So they're quite correlated, even though they're distinct categories. And to answer my second question, so you remember we've got these 6 domains of adaptive capacity. And from these 6 domains, there was 33 questions from the survey. And from the answers of these questions, I summed them together, normalized them, and created 6 indicators for each of the 6 domains, well, 1 for each domain. And from these, I compared them via ANOVAs for each type of social organization across independent, cooperative, and patron-client. And for the last question is how they're responding differently to climate change impacts than other types of social organization. So here we have all of the responses that they did, and they were categorized into 3 types of responses. So economic, fishing, transformative. And I identified all of the ones that met the requirements for the statistical tests I wanted to do, because some of them were quite low numbers. So all of the ones above the dotted line were selected for further analysis. And I ran chi-squared tests on all of them across the different types of social organization. This might seem like a result, but it's a method, I promise, because it goes into what I use for the model further. So all of these significant tests within economic, fishing, and transformative, I then used within my multivariate probit model. So the significant within fishing, those 2 were included in fishing, and economic was borrow money, that was the only significant one, etc. I also included the 6 domains of adaptive capacity as well as social organization. So this is the differences between independent, cooperative, and patron, which is what we're interested in. So, results. For the first question, so what type of social organization exists, we can see that the majority of fishers are within patron-client relationships. And why we think this is because there is such a shortage of permits. This is the main route for fishers to access permits. And they're so important because you can get bursaries from the government if you have a permit and you don't have to fish illegally. However, this doesn't really reply to why cooperatives are so low, because cooperatives technically provide permits as well. But previous literature has shown that fishers have complained recently about the cooperatives because from the 1990s in this reform that I mentioned at the beginning, the number of people required to make a cooperative has changed to 5. So this means that there's been a lot more family-based cooperatives, and fishers have been sort of disgruntled by this because they think they're no longer proper cooperatives and they're not serving their function, and they're more like family-based patrons. So we think that's why there's lower numbers here. And for the second part of the question, you can see in this graph, the majority of people in patron-client relationships borrow all eight benefits. So this is quite a high level of dependency the majority of fishers have on these patrons, and kind of increases our thoughts on this power imbalance between the patrons and the fishers. So for the second question, I just want to highlight these values here are the significant values for the ANOVAs that I ran, and you've got the 6 domains here across independent, cooperative, and patron for each one. And the first thing I want to highlight is that the lowest for everyone overall for fishers was assets and flexibility. And within assets, the key indicators were income and access to governmental support. So previous literature shows it's no surprise small-scale fisheries have low incomes. It's not a surprise at all. And the second thing, if, like I previously mentioned, you don't have a permit, you can't access governmental support. So the fact that everyone scores lowest in assets makes sense. And secondly, for flexibility, we think that there's not only a high level of dependency economically on small-scale fisheries, but also socially as well. You know, this is a huge part of people's culture, of music, of art. It's not just something that they do for a job, and it's been in people's families for generations. So, it's specifically about those in patron-client relationships. They significantly scored lower on assets, and we think the key indicator here was boats. So, previous literature suggests that patrons often provide boats. We saw this in the research and on our interviews as well, and that's maybe one of the main reasons why fishers are going into these relationships with patrons. And the second result here is you can see that the order of agency scores goes independent, cooperative, patron. This, I don't know if you remember, kind of mirrors our scale of autonomy that we hypothesized, with independent fishers having the highest, followed by cooperatives, and then patrons. And one of the key indicators here, no surprise, is having access to a permit. So we also saw this within the survey results as well. So independent fishers had, 36% of them had access to their own permit, whereas fishers in patron-client relationships, only 6% of them had access to their own permit. So this is reflected here. So it makes sense. And finally, this is my final question. There's a lot of numbers here, but just sort of focus in on this here. This basically means that fishers in patron-client relationships were more likely to adopt economic responses. And this is backed up by the chi-squareds that I ran and you saw earlier, where you can see that fishers in patron-client relationships are more likely to borrow money than independent fishers. And we think that this is because fishers have a lack of access to formal money lending. So patron-client relationships might be the only way that they can borrow money. So this might be why that fishers in patron-client relationships have higher levels of borrowing money. And although this is quite good for short-term stabilization of the small-scale fisheries, it can also result to quite bad adaptations as well, because we also see that fishers in patron-client relationships are more likely to increase their fishing area, so they want to work off the debt that they have with these patrons, and start illegal fishing. And interestingly, what we saw noted in the interviews and also in previous literature is that some patrons have been known to encourage illegal fishing practices. So the fact that we see here they're more likely to start illegal fishing is in coordinates with the previous literature, and isn't really a surprise. And finally, we see that those in patron-client relationships are more likely to increase their alternative livelihood and migrate. And from previous literature, this kind of makes sense because we see that those with more assets, more physical assets, they've been seen to be less likely to want to migrate. And this makes sense because if you have a boat and a permit, you're not really going to want to move city. And also, if you are already really invested, you're not going to want to change your livelihood because you've already put so much into it. And lastly, those that have so many assets in the first place, some previous studies have shown that they feel more confidence in their ability to sort of stand in the face of climate change and in the changes that are coming. So because they have these assets, they think, I don't think I need to, I think I'm going to be okay. So they don't feel the need to do these transformative responses so much. So, to conclude, 3 main things to take away. Most fishes within Nayarit are in patron-client relationships, and they have a high dependency on patrons. Fishes overall scored lowest in flexibility and assets, with fishes in patron-client relationships scoring lowest in assets and agency. And then finally, fishes in patron-client relationships are more likely to respond overall to the changes due to climate change. And this is really important because there's been a lot of worry about the sustenance of small-scale fisheries, and they're so important. So we can see here 2 things that are really important where we know that they can be helped and how policy can be focused. And with climate change, we don't know how they're going to be able to respond, but we can see clearly here that they don't all respond in the same way. So this needs to be taken into consideration. And for future research, I have a lot of ideas, but the main one that I want to concentrate on is interviews with patrons. So the previous literature shows that patrons, they respond differently as well. They're not all the same. And based on their links with other people can change how they respond to climate change as well. So interviews with patrons would be really interesting to see their side of it and how they connect together. So I would like to thank everyone on here, but especially my supervisors, Sachila and Elena, and thank you to all of you for listening. I hope you enjoyed.
Thank you. Within the minutes, perfect. Anybody want to start with the questions?
I can start.
Yeah.
Thank you so much and congratulations for all the interviews and all
Thank
the
you.
And now the question, did you design yourself the questions you asked for them?
So it was my supervisor, she's doing her PhD, so she designed the questionnaires and she completed the questionnaires because they were done in 2021. So that was before I even started the masters.
Okay. And along the time you were doing the interviews, you changed questions or they were all the same?
No, so the questions were based, we have a huge list of the literature that they were based upon, and so 33 of them were based on the indicators for adaptive capacity, but a lot of them were just socioeconomic questions like how much do you earn per week, what fish do you use, what gear do you use, that kind of thing. And they tried to keep it pretty sort of uniform across the months, and they had prepared an interview which they used the whole time.
Okay. So they were all the same for the people interviewed?
Yeah.
Okay. You think you should change when you interview the partner in the...
Yeah, definitely. There's so many different things that you could ask. Yeah, and even if I was to do it, well, if we were to do it again, there's something else that I wanted to mention on the conclusions that, so here we sort of assumed that high levels of engagement meant that they had high levels of dependency on patrons, but that could also be interpreted as if you have a high levels of engagement with someone, that means that maybe you have a strong bond with them. So maybe in those relationships you have more flexibility because you see them more often as opposed to someone that only borrows a permit or something like that. So I would include those kind of questions as well as their perception on their dependency of the patrons too.
Just make it more informative.
Yeah.
Okay, and just the last question. During your presentation, you always explain like this is not surprising. So do you have any result that you say, this is surprising me, I was not expecting that from the interviews or from...
Well, it wasn't surprising when we looked at the previous literature from other places, but I forgot to mention at the beginning that there has been no previous studies like this in Mexico. So we didn't know what we were going to expect. But it kind of, it's nice because this means that it's in accordance with other places, so it can be sort of extrapolated and used for guidance as well too.
Okay, thank you so much.
Well, thank you very much, Sarah. It was a really nice presentation. And this is a fascinating topic. I think that there is a lot of, I don't know, it seems like the way of solving the issue with fisheries in general. You know, who should make the decisions, who should have the power, and you know, there is a lot of thinking and a lot of things moving on in that direction. I must admit I have some, you know, some difficulties and struggle a bit to follow some of your results. So I have an evil question. In this case, what is a multivariate probit model?
Okay, so basically I chose this one as opposed to just a normal probit model or a nomial model or anything like that, is because it takes into consideration, multivariate means that the fishes can choose multiple responses at the same time, so it's not always just fishing or just economic. So I chose this model because it can incorporate multiple responses at the same time, and they're not leveled, they're independent responses. And that means that it takes into consideration the independent variables that you put in as well. So we wanted to see whether or not the domains affect the choices that they choose, and as well as the types of social organization.
So for instance, in the last graph that you showed,
yeah,
I had the impression, you know,
these ones?
This is a naive question. So from that graph about the start illegal fishing, that means that about quarter of the fishermen enrolled in a patron fisherman relationship will be willing or will start to do illegal fishing, and that probability doubles.
So, sorry, what's your question?
Can you, for instance, interpret for me
the graph here?
Yeah.
Oh yeah, okay. Well, so this isn't from the multivariate probit model. This is from the chi-squads.
Okay.
So I ran chi-squads across the 3 types of social organization, and it basically means, you can also see as well, so in between independent and patron, patrons are significantly more likely to conduct sort of illegal fishing.
No, I mean the level.
The level.
How
Well,
do
but
you
it's not.
interpret
I mean, you
that
can't.
percentage?
I mean, you can, but it's not part of the test. Like, you just know that they're significantly different because of the grouping, but it's not like, I mean, you can see here, so it's like you've got, I don't know, maybe 12% here and then 25% here, but from the test, you just know that the groups is significantly different, not the proportion.
Yeah, but for people who don't believe in tests, you know what I mean?
Yeah, okay, so maybe 2 times. You can see it's maybe 2 times more people are likely to do it.
What do you think about it?
I think it's interesting because there was something as well in the interviews that the fishers said that there's this type of fishing that requires a lot of people that independent fishers can't do, and sometimes the patrons in Nayarit were accepting of this. So this can maybe explain why people in patron-client relationships are more likely to do it, because if the patrons are sort of organizing it and accepting of it, then that makes sense. And I also think it's like a social ecological trap, you know? Like, they're borrowing more money, so they're in more debt, so they need to get out of it, so they're increasing their fishing effort by whatever means is possible. And I also think it's kind of unfair because there's not a lot of permits, so a lot of fishers are forced to kind of fish illegally because there's not a lot of permits to go around.
Okay, I like it, but you know, I miss it a little bit more on that kind of interpretation. You know what I mean?
Yeah, I mean, I would love to talk more about it, but the time.
Okay. Anyway, thank you very much.
No, thank you.
Yes. Yeah, I think you had very nice and clear slides. You took the risk to lose 20% of the surface area with your fishing.
You didn't like the fish?
No, but they reduce and then you are obliged to at the bottom the question, which is squeezed to the bottom and not bigger. And so, but I think you made it quite well, and you are lucky that the screen was quite high, so I could see. So yes, so that's good. So my question is, what do we know about the impact of these 3 different organizations on the fishing efficiency?
What would you mean fishing efficiency?
Which one provide or catch more fish per unit of effort? Is there a relationship there or not?
Are there regulations as to how much each organized
Is
using
there
fish.
a relationship between the type of organization that is chosen and the amount of fish that you provide to the community per unit of effort?
That's a good question. Within the interviews, there were questions on how much do you fish per week and that kind of thing, but that I didn't go into that, so I don't know. Can't answer your question.
But do you think there would be, or is something more efficient? Is a cooperative when people maybe talk more together, then they know where are the good spots, so they would help each other, or?
In the domains of adaptive capacity, some of the indicators were like, do you communicate a lot with other fishers, and do you communicate a lot with other people in the community? And cooperatives actually scored quite highly in that. So I imagine that they speak a lot more. So potentially, maybe they do know where the better fishing spots are. But however, you could argue also that fishers in pigeon-client relationships often work in groups in their boats. So it's a tricky one. I don't know. I don't know what the answer would be.
Okay, and then so to fish whatever organization you choose, you need boat, you need petrol, you need ice, as you said. In the patron organization, if I understood well, it's one fisherman and one patron.
Yeah, so when you're in a
And the patron is taking all the risk because he's buying boats, he's equipping the boats, so he's borrowing money at the bank, and he doesn't have the money.
So your question is?
So I don't really understand this idea of borrowing money. I mean, still everybody needs to have money to make a boat.
Yeah.
So all the groups, all the different structures, they do take this risk. So I do not understand why you make a difference here.
In between the types of fishers that borrow money?
Yeah.
Well, so I don't know. I'm still not sure if I understand your question.
So the patron owns
Yeah.
the boat.
Yeah.
He has to buy the boat.
Yeah.
So he is taking a risk when he's buying the boat.
Mm-hmm.
The same way that the cooperative is doing.
Yeah, but these aren't, maybe it's unclear, but these are fishers that work independently within a cooperative and within patron-client relationships. This doesn't mean patron. It means those within patron-client relationships. So the fishers within patron-client relationships are more likely to borrow money because they have the patrons to borrow it from, whereas independent fishers have lack of access to formal money lending.
Who is the patron?
So a patron can be anyone that has access to a permit or has access to a boat. They often are able to transport the fish to bigger markets that the fishers themselves don't have access to. So sometimes they're in city of Mexico. So the fishers themselves don't have the transportation to take them, so they will sell them to a patron, which will then sell them on, or they'll work underneath a patron.
So what does patron means in English? Because in France it has a very specific meaning, so I was confused a little.
Patron is just patron.
It's a boat.
of the boat.
Yeah, well, but sometimes it's not always the captain of the boat. Like
when
The owner.
He's a owner.
Like the owner.
in Spanish we call it patron. It's the capitan, the armador. It's the one that owns the boat, and then the crew is the people that works in the boat. Also, you can be the armador, the owner, and also the capitan of the boat. Sometimes you share boats, roles, but not necessarily.
Yeah, that's right. Thanks, Alex. So you can sometimes be just the owner, but usually it's someone that has access to the permit, because that's the main reason why they're entering into these relationships.
She's referring to the relationship more than the.
Okay, thank you.
No worries.
Can I ask you, so in the context of ever scarcer resources, I don't really see the positives of this patron fisherman relationship, because, as you said, it increases the fishing effort and stuff like that. Some benefits, you said, some positives could be the adaptability to climate change and stuff like that. But I'm thinking that in terms of management of fisheries, and nowadays everybody's pushing for bottom-up approaches. How do these three different, is this going to facilitate bottom-up management? But this has to, compared to independent fishermen or fishermen integrated in a cooperative system? How do you see it?
Yeah, I'm glad you asked that question. So because the majority are within patron-client relationships, then it's not all negative. Like, sometimes fishers can't afford to have their own boats and they don't have access to permits. So with patrons, they have a livelihood. So yes, sometimes the adaptations are negative, but it's not always like that. And the majority of the time, they are providing for the development and the growth of the fishery. But I think what's really important is that the majority of the time, the policies are focused on the fishers, right? They're like, you can't catch above this size, at this time, at this place, but you have this second level of force that's pressuring them to fish at certain times because the patron decides what species they want and at what price they're going to buy it. So instead of maybe regulating the fishers, in places that you identify that there's more people within patron-client relationships, maybe it's more effective to focus on the patrons instead because they're the ones kind of setting the limits and telling the fishers what they want. So I think they can be really beneficial in the sustainability of the fisheries as well. And their links and their connectivity with each other is a really good tool for communication and for spreading new things, and maybe in the face of climate change, they can be really useful to spread information as well.
Any questions?
I understand you don't have time to go into it, but what's the reasoning for this lack of permits? Like, is it overfished or?
So the permits are set by an organization within Mexico. They set the number every year, and it's based on like catch levels, but the catch levels aren't accurate in the first place. So the amount of permits is set on a false number to begin with, and that's why the reason why they're so low. And we already know that they're being overfished, so I think the idea is to try and limit fishing via the permits, but it's not working, and it's not been working for a long time.
Just to comment that without considering myself an expert in fisheries, I would say that more or less is similar everywhere. So I think that the applicability of your work would extend well beyond the problems that you are analyzing in Mexico.
Yeah, when I was doing my sort of literature review at the beginning, patron-client relationships are really common within Asia and within Africa as well. So I think this kind of policy should be really looked at and focus maybe more on the patrons when we're trying to look at the sustainability of small-scale fisheries.
Well, and just to add on to that, in West Africa, countries like Senegal, for example, it really exasperates, worsens the situation in terms of overfishing. We're seeing where these patrons, they provide the boats, and then they bring in these fishermen and pay them miserably, and then they have to work like slaves, and it really adds to worsens the situation in terms of overfishing.
Yeah, it can be very
overexploitative. They come there, spread out along the coast, and these patrons drive along. They bring the ice, they bring the catch, they bring them the food. They're totally dependent on them. So.
Yeah, exactly. Which is why I think it should be more focused on them instead of the fishers in some places where we identify this kind of thing.
Okay, I see no questions on the chat. So, thank you very much.
Thank you.
Nice talking to you.
Yeah, well, I was stressing out with that, you know, I was able to disconnect.
In 300
Okay. Yeah.
Good. Absolutely. Now I understand what you were doing. Before I owned it out. But it's good that I understand at least now.
Wow, at least later.
Better late than never.
Obrigada, Doreen.
Nada.
acción, ¿verdad? Vamos a mover entre los primeros. ¿Quieres ir para acá? Pero vamos a mover igual entre los primeros.
Necesitamos dos punticas para que, ¿verdad? Pero bueno, nosotros somos cuatro.
Hola. How are you? Good, good. How are you? This is my family.
Hi, how are you? Welcome.
How you doing?
Good, good.
You're presenting right now?
Yeah.
Nice.
You got it. Presenting first. So.
Ooh.
And then our use and then you.
Setting the mood. Like it.
Yeah.
Nice.
It's nice because then you can chill for the rest of the
Yeah. I mean, yeah, I'm not here to stuff you, but you'll do it. So, and you're always so calm. So I have no
have to speak a little slow. I'm speaking a little slow. Yeah, it's a little bit wings for Oh, can we have you checked your presentation yet?
Oh no, it's not.
I want to see if everything's in order for you.
Hi. Is it okay if you see that our presentation is in order?
It's 3 and yeah, we have to go to 4 anyway.
Ah, perfect.
Of course. Will you share the session with me? I will.
That's very good.
Okay.
I also knew I was in the right room because I saw you here.
Okay.
So,
Okay, this is good. This is
also for the
good.
timing, will you give any indications?
Yeah,
that's
try to wave at you like 2 minutes to
Okay.
go.
2 minutes,
Alright,
okay.
yeah. Okay.
But that means you have to look this way every now and then,
Yeah, yeah.
or that's the only thing.
Okay, everything is in order for me. So I'll just go ahead and close it. Hello.
Hello.
Lars,
Hi, Lars.
You want to check yours or?
Yeah. Wait, I need to fix my lip first.
Will you have your hound here?
Well, no, it's not the very smooth hound, unfortunately. But it is this mini dogfish, so it's, I think, close enough.
Oh, it's also with Wageningen.
Yeah.
Just so you know, it looks like it was left streaming from the last. So this is all.
So they can-
This is all on YouTube. You can wave it around.
Okay.
It's still streaming, yeah. It doesn't matter, but just so you know.
Okay.
And it'll all be teased.
I would like to. Oh, there's no Acrobat reader.
Somebody left their note.
No, that's mine.
Oh, this is yours. Okay.
This is a map of Belgium. How do you? Oh my god! How do you say full screen view here? Hello.
Hello. Yeah, it's still streaming. It's still on YouTube, so.
Oh, you
Do
got
you
it.
know? Yeah,
We're-
but
we're live at the
so
moment.
I have a question. I have a PDF and the slides. They are firstly
It's just gonna load a little bit slow, but it won't.
okay
It
because
won't
it,
affect
it it didn't do that on like it doesn't do that on my PC.
Yeah. It's just something on the resolution of the screen, I guess.
Alright.
If you go to the second slide.
Wait, sorry.
Are you presenting as well? Yes. Any videos?
Sounds
Yeah,
kind of.
I had PDF in here as well this morning, and it wasn't doing that, but it will have no reflection on.
Okay. Sad though, but
Yeah.
okay.
Oh, but you can still see just this is
Of
a
course,
second
It
version.
just
Yeah,
takes a
but I
second.
mean-
Yeah. You can be keeping people on their toes that they're paying attention.
Oh yeah, that's true. Can I select it? Sure. Because then we don't have to stand. To be fair, I have an image in the background, so probably like the pixel resolution is very high.
I think we could just lower the brightness
Oh, that'd be nice.
because it's too.
It's very bright.
Too light, yes.
Oh yeah, that will make it.
Yeah, it's way nicer.
Oh,
Oh, I should have told the people we were here last session and they also had issues
ahora se
So, but these two should be fine, right? We can leave the ones in the back open.
I feel like, I mean, I'll try, but I would.
I think this one's done.
This is broken.
Yeah. No vas más de ahí.
Like here, there's no.
Well,
Ah, okay.
it's the other way around.
No, sí, sí, porque estoy yendo a la cuerda. Ah, sí, ahí se ve mejor.
Oh, yeah,
This was
maybe
way
I
nicer.
could-
Yeah, okay. Maybe we should still go on the first side. So Carmen, will you be sitting somewhere here?
No,
okay.
yes,
Oh, right.
moving between containers. But I want to make sure that all the can I escape? Yeah, of course. Go ahead. Okay. Wait, so you're the first, and then
Norvis and then any other.
okay, and it's just 3.
Yes. I mean, it's already 5, so it's good that it's only 3 presentations and we're not here until 7.
Yes.
Yeah. Get this out of it. Go fly. Where's the second speaker? Oh, yeah.
No.
How is it? Second. Yeah. How do I pronounce that? Nice. Yes. I don't want to completely yes. Okay. If are you happy to start anything?
Yeah, it's
Okay. There's still 2 minutes. We can
I
wait.
mean, the sooner wait. Yeah, the sooner coming.
Yeah. Then we're happy to wait if you're happy to wait. But you're
Yeah, I'm happy to wait.
Cool. It's from this morning. Somebody had started this morning. So maybe I actually planned for that. A mini one. Spontaneous. Yeah, yeah, see what happens. Sounds good. And then Friday will be
nice.
less than spontaneous. That's great. Good. Okay, so I'll start welcoming everybody. Okay, so welcome to our final session of today. We have 3 speakers in this session. Each speaker has 15 minutes to present, 15 minutes for questions from the jury and the audience, and also if there's any questions online, and I see there's some people watching online already, so welcome to them too. I will give the speakers kind of a 2-minute wave, but if you're not looking in this direction, you might miss it. So if you want to just scan every now and then, I'll give a 2-minute wave, and then otherwise I'll stand up if it's really going over time. And that's it. So our first speaker is Lotte Pohl, who's going to talk to us about starry smoothhound sharks. So I'll leave it to you, Lotte. Best of luck.
Thank you. Yeah, thank you, Katie, for the introduction. Welcome, everybody. Glad to see so many of you here. My name is Lotte, and I will talk you through my master thesis that was about the movement and seasonality of the starry smoothhound shark, Mustelus asterias, in the North Sea. And I conducted this thesis at the Flanders Marine Institute in Belgium. And I would first like to thank my supervisor, Carlotta, and my promoters, Jan and Elias, for guiding me through this thesis. So marine animals frequently move between jurisdictional boundaries, such as exclusive economic zones, as well as beyond waters that we can easily access, like coastal waters, and then they move into the open ocean. And this makes it very challenging for us to track their movements. However, tracking animal movements is very important because understanding the how, when, and why related to animal movements helps us to improve our management and conservation measures of highly migratory species. My species of interest, the starry smoothhound shark, also exhibits some complex migration behavior involving annual migration and having a summer habitat and a winter habitat that potentially differs between sexes. So Mustelus asterias is a very frequent shark in the Northeast Atlantic Ocean, inhabiting waters in the southern North Sea, the English Channel, even down to the Bay of Biscay. And previous studies suggest that at least one population resides in coastal waters off of Belgium and the Netherlands during summer, and then males go up north to the east coast of England during winter, whereas females go south to the English Channel and the Bay of Biscay. But the seasonal presence of the species in Belgium and the Netherlands has only been recorded in the last 15 years, so we don't really know that much about the details as to when and why these animals are in these waters. And moreover, the animal migration is also not very detailedly studied, and details lack due to concerning the timing of the migration, as well as if really the winter habitats between both sexes differ. So with these points in mind, we wanted to assess the migration behavior of the species concerning two scales. Firstly, the local seasonal scale, addressing the presence of the species in Belgian and Dutch waters, and secondly, a more broad scale addressing the annual migration patterns of the species. And for each of these research lines, we wanted to look at seasonality, so whether or not movement patterns differed between seasons, and then secondly, sex specificity, so whether or not those patterns differed between males and females. We tracked the animals' movements using so-called acoustic data storage tags. And those are novel electronic tags that combine two very popular tracking technologies, namely acoustic tags and data storage tags. So for acoustic tags, those tags are implanted into animals and they emit unique acoustic signals that can be picked up by an acoustic receiver when the tag is within a specific range of that receiver. And those acoustic receiver networks are generally maintained in coastal areas that are accessible to humans because those acoustic receivers need to be submerged and also be accessed by humans to download detection data and to change batteries. So as we move offshore into deeper waters, less accessible to humans, data storage tags come in handy. And those tags continuously log temperature or depth or other environmental variables. And we get these very high density data logs only upon retrieval of the tag. So due to this dual technology nature of the tag, we were able to address the two scales of our study with these tags, namely the local scale concerning the seasonal presence in Dutch and Belgian waters with our acoustic detections, and then the annual migration patterns with our data logs. And I will be focusing on the depth logs here. Tagging occurred in 2018 and 2019, and 30 individuals were tagged. And you can see some photos of the tag implantation procedure here. Each tag was active for about 18 months, logged depth in 2-minute intervals, and the depth sensor could measure up until 70 meters of depth. Concerning our acoustic detections, we utilized the permanent Belgian acoustic receiver network that is comprised of about 160 acoustic receivers, and each of these receivers is represented by a black circle on this map. You can see the two tagging locations in yellow, and I would like to draw your attention to the scaled estuary, which is an estuary in the Netherlands comprised of muddy and sandy habitat. And it's divided into the eastern scheldt on the top and the western scheldt on the bottom. And especially we have 3 receiver gates, so receiver lines marked in colored rectangles here that allow us to study animals moving in and out of the estuary. So because we have this dual nature of our tags, we also have 2 types of data sets that come out of these tags. And firstly, we analyze the acoustic detection data by looking at the detections per individual and where and when those detections occurred to infer seasonal patterns and sex specificity. And then for our data storage logs, we wanted to infer underlying recurring patterns to infer behaviors and those analyze those regarding to sex and seasonality. And we did this with wavelet analysis, which is a type of spectral analysis, and I would like to briefly outline this analysis to you. So imagine you have a time series signal with a recurring pattern, let's say sunlight intensity, for example. Then the time each cycle needs to complete itself is called the period or the cycle duration, marked in red here. And for sunlight intensity, this would be 24 hours. If we now consider the depth time series of a fish swimming to the water surface and then back to deeper waters once every 12 hours, then the period would be 12 hours. But now we imagine the fish changes its behavior to swim to the water surface only once every 24 hours. Then, as you can see here, the period would be 24 hours. So now, if we do a wavelet analysis of this simplistic exemplary signal, then we would obtain a wavelet scaleogram like this, where you have the period on the y-axis, the time on the x-axis, and the colors represent the spectral power, which is a proxy for the dominance or the prevalence of that period in the time series signal at each point in time. And we watch out for these highly red colors because those represent highly dominant frequencies. And if we now look at these 2 red colored bands, those represent our 12 and 24-hour period band if we look to the y-axis of our input signal with the 12 and then the 24-hour period. But coming back to the acoustic detections, 18 individuals were acoustically detected in the receiver area, and we obtained about 11,000 detections over a rough period of 2 years. And interestingly, 97% of detections were from females, and 97% of detections occurred in receiver array number 1, marked in orange here, which is the outermost receiver gate in the Western Scheldt estuary. If we now look at the detections per individual, we have the individual number on the y-axis and the time on the x-axis, and from this plot we can see 3 things. So firstly, each colorful circle represents 1 acoustic detection. And we can see that all detections occur within April to November. So this indicates seasonal presence of the species in the receiver area. Secondly, we see that many individuals were detected in consecutive years, indicating site fidelity of the individuals. And thirdly, we see that most of the colored dots are orange, indicating or representing that the detections happened in this receiver array number 1. So we have seasonal presence from April to November, we have site fidelity, and we have a strong sex specificity or sex bias towards females. About the recovered tags, 8 tags were able to be recovered out of the 30 tags that were deployed, but only 2 of the tags logged data for over a year, allowing us to investigate these annual migration patterns in the vertical space. And we have the depth log of 1 female and 1 male here, and the raw depth is colored blue, and the black line represents median daily depth. And we see somewhat similar patterns between the 2 individuals, with shallow depths between 0 and 20 meters occupied during summer, then a steep depth increase during autumn, and then deeper depths occupied during winter, where the female went much deeper than the male to waters of 50 to 70 meters and the male only to 20 to 40 meters. And then they have both a stepwise depth decrease until reaching those shallower summer waters again. So now, if we look at our wavelet scalograms, those look a bit more complex because now our depth signal is not only 1 harmonic frequency, but instead a superposition of many harmonic sine waves with different periods. And we are interested in the prevalent periods here. And if we look at the female scaleogram to the left, we see that it has some prevalence of the 12-hour period, indicated by slight orange colors. And then for the male, we also see that on top of that, it has strong prevalence of the 24-hour period band, indicated by red colors. So that already marks a difference. If we now look at the winter, then we see that the female has both prevalence of the 12-hour and the 24-hour period band, but also has strong prevalence in the 2-week period band. And if we look at the male, it has some sort of prevalence of the 24-hour period and also prevalence of the 1 to 2 week period bands. And now, explaining this, we can look at a subset of the summer depth log of the female here on the left, and we see that it mostly displays a tidal signal. So imagine the fish lies on the ground and the water rises and falls as the tides go in and come in and go out. And because it's 12 hours from one high tide and to the next high tide, this is a 12-hour period, which explains the 12-hour period prevalence that we saw on the scaleogram. Now for the male, it looks slightly different. And the male seems to go to the water surface every night, so when one day changes to the next. And this indicates diavertical migration, which was linked to feeding behavior by previous studies. And because it's 24 hours between one day to the next one, we can explain this 24-hour period band that we saw in dark red colors in the wavelet scaleogram of the male. Now, if we look at the winter depth log, we see that the female kind of seems to rest on the bottom at 60 meters roughly for some time, and then displays very high vertical behavior after some time, so using a high depth range. And this pattern reoccurs about every 2 weeks, which is interestingly potentially related to moon phases, which in turn influence the strength of tidal currents. For the male, on the other side, we were not really able to detect any emergent behavioral patterns from the raw depth log that might explain the prevalent periods in our scaleogram. So this requires further investigation. But before concluding, we need to, of course, discuss the methods and their limitations. So we implanted tags into the animals. So those tags most likely have an effect on the natural behavior of these animals. And for now, unfortunately, tag weight and volume have to be taken into consideration with the battery lifespan. So there's a kind of compromise that needs to be made. Also, the handling of the animals, so taking them on board and performing the surgery, of course, stresses the animals. So that just has to be taken into account as well as try to be minimized as much as possible. But looking at our acoustic detections, we can say that 11,000 detections are already a starting point, but looking at other acoustic studies, it's a very low number, which needs to be increased in order to make more robust conclusions. Then we also had mostly only detections of females, so we don't really know where the males are. And then our gate-like receiver setup, which you can see here again, is a bit suboptimal when studying or when wanting to study the horizontal distribution of a species in greater detail. So yeah, this is just a limitation we had to work with because of the acoustic receiver network. Concerning our recovered tags, we only had 2 tags that logged data for over a year. So now we are not really able to say if the differences in vertical movement are due to individual variation or due to sex. So we need more depth logs in order to further investigate this. And moreover, our depth sensor had a limit of 70 meters, but you can see a subset of the depth log of the female, and it most likely obviously went deeper than 70 meters, so we don't really know which depths the female really occupied. To conclude, we were able to confirm the seasonal presence of mostly females in Dutch and Belgian coastal waters for the first time in the Western Scheldt. And this might be related to pupping behavior. And Brevet et al. Already proclaimed a pupping ground in the Eastern Scheldt, so the adjacent estuary. So this is interesting to further study. And then concerning our annual migration, we could see seasonal differences in the depth logs. So shallower depths occupied during summer, deeper depths occupied during winter. And we could also indicate some potential sex specificity because the female showed more resting behavior during summer compared to the male who displayed more diavertical migration during summer, and during winter both individuals displayed diavertical migration and the female also displayed some resting behavior. Concerning spatial preferences, we cannot make any direct inferences from our vertical depth log considering the latitude and longitude of those individuals, which would need to be done with geolocation modeling. So our next steps would be to investigate the potential pupping behavior further for this species in the Western Scheldt estuary, and also to use the outcomes of our wavelet analysis to conduct geolocation modeling to finally infer latitude and longitude from our depth logs. And you can see an example of that here on the map. And with this, I would like to thank you all very much for your attention, especially those people on the slide. And yeah, I'm open to any questions. Thank you.
presentation and very well timed. Questions from jury members first? Yeah.
Thank you for a very nice presentation. Very clear. I think the way you explained the wavelength analysis it was very, very good because I understood clearly.
Oh, that's nice.
And I know it's not always an easy method. You mostly observe that the females do respond on with a tag. And you propose directly that they come for cutting. And then you don't say a lot about the male, but what could the literature tell us about the males? Where are they? What are they doing?
Interestingly, studies about this species in this area have only started 10 years ago, and then most of the studies were conducted in the British Channel, so not even in this area. So my supervisor, who conducted most of all of the studies that were in this area, has no clue. There might be a slight overlap in habitat in the Scheldt estuary, because we had some detections of the males in the Belgian part of the North Sea, so not within the estuary, but in this small part of the North Sea of Belgium. So it's highly likely that they are in the area, because also the two sexes need some sort of overlap to potentially mate at some point. But the general belief is that the males just stay more offshore, and they don't move into the coastal waters where we can pick them up with receivers. But as to where in this broad area they are in summer, nobody knows yet. So we need further studies on that.
Where do they encounter the females to reproduce?
Well, mostly in the estuary itself, and so at the outer part of the estuary.
How can you be sure they meet in the estuary if they do not share the same depth level, and even the male don't really go to the estuary?
Yeah, no, we don't really think that they meet in the estuary, but more like in the realm of the estuary and around it. So, for example, in the Belgian part of the North Sea. So we know that females, they tend to stay in the estuary, but also what I didn't mention, which makes matters a bit more complicated, is that the species has a biannual reproduction cycle. So we don't really know if females, if they are in their pause year, let's say, if they also go into the estuary, or if they only go in there to pup. So it could be that some females don't go into the estuary, but more stay in the coastal waters, where they then can mate. But mating behavior is also very very hard to study, because the species can store sperm for over 12 months, and they can fertilize themselves. So we didn't find any way yet on trying to find out where they potentially mate. But we basically need to tag more males, but also because the tagging is done in this coastal part, there's in the tracking studies so far, there's always a bias towards females. So it would be interesting to really target the males to find out where they go. Yeah.
Maybe another question. So you also show that females tend to stay at the same depth, but the water layer is changing, while the male are migrating. I did not understand if they go at surface by night or by day. And what would be the reason? You said to pray, but
I try to go back, but it's a bit slow. So with this rising and falling water, I just wanted to here we go. I just wanted to explain this the sinusoidal signal, which is the tidal signal. It's just basically the signal of the tides rising and falling. So the female is not moving at all. But here for the male, you can see, maybe without, for the male you can see that, so this is when a new day starts, this solid line. So when a new day starts, so at night, the male here goes up to the water surface. And because it is a crustacean specialist, previous studies proclaim that the male goes up following the diavertical migration of plankton and then crustaceans, so then the males also go up. But interestingly, the females, they don't go up, which might be because they are gestating, and many migratory fish, they stop feeding when they migrate to their pupping habitat. So this could be an indication that this female, only resting at the seafloor, is gestating, but of course this is only a speculation at this point.
thank you.
Yeah. So first, congrats, you're here. I mean, and I agree that you explained really well. I mean, even for me, that I'm not an expert in this, was super easy to follow. So that's really, I have to appreciate that. So well done. You explained really clearly. I think it's a nice study. Of course, all the study has some caveats or some limitation, and I think it's quite nice when you acknowledge those. And I think it's a way to go. No, it's never hiding limitation. It's always showing and to say, okay, consider this limitation. I think that we'll find this and that. Of course, yeah, the data you have, it's one individual versus the other, and as you acknowledge, but it's a starting point, and I think it's great that, and it's good to highlight. And of course, maybe, those differences, you didn't have more individuals. The other thing is, I guess I know the answer, but just I want to ask you, why you have a bias in the tagging? Yeah, because I think it's 14 females versus 4 males. Knowing that there's scarce information about males, why you didn't try to do the opposite, because you couldn't find it, or why? Can you explain that?
Yeah, of course. So first of all, yes, there is a bias towards females in tagging, but we tagged, or not I, but my colleagues tagged 19 females and 11 males. So the bias is a bit smaller. But of those 19 females, 14 females got detected, and of the 11 males that were tagged, 4 males got detected. So you already see that, considering the bias, but the proportion detected is much smaller in males. And this was a more exploratory study that was done. So the overall game was to use this new tracking technology and see if it would get viable results. So the people really also only wanted to confirm the seasonal presence of the species is in the Western Scheldt, because it was not studied before. My promoter had done some work in the Eastern Scheldt, but not in the Western Scheldt. So I think this would be a next step to especially target the males. But for now, they basically went out fishing, tried to fish sharks, and they got more females than males. And they even encountered 2 neonates while tagging. So this, of course, is also an indication. So yeah, but this would be a relevant next step, because you need to know where the males are.
Then the last question I have is, can you somehow obtain some data from fisheries? Like, these particular sharks get caught in the trawl, and then, for example, you can see, okay, the position, even if they're present, or if the male distribution. Do you have some sort of information or like that, or there's not?
Yes, there are ICES trawling fishing data, and previous studies also already looked at the species, but more in the UK waters. But they did use those data to look at if they found differences in distributions between males and females. I'm not particularly sure, for example, for the Scheldt estuary. Inside the estuary there's no real trawling going on, but of course for the coastal waters it would be interesting to look at those trawling data and see what you find. But in general, I think it's very important to include the collaboration with fishermen in these studies, because only they, for example, highlighted, oh, we catch these fish now, we didn't catch those before, and that kind of sparked the first study of my promoter in the area. So I think generally collaborating with people that know the fish and that work in these areas is super important. But yeah, definitely it's a nice study already, but it is a bit data scarce. So in the future, different data sources should be tried to be sourced in order to get to more data-rich conclusions, definitely.
Okay, once again, congrats. Thank you very much.
Yeah, yes, a quick question. Thank you very much for your presentation. Related to Gonzalo's question, do you have other methods that are not so invasive, except from tagging?
Yes, I've just been to a tagging conference. So one person just presented a new method, which I'm not entirely sure if this shark is particularly good, suited for that, but you could attach tags. Well, first you can attach tags outside at the fin. This is usually done with mark recapture studies. So you mark the shark, and when if it's captured, then you log the recapture location. But those external tags, they often are highly biofouled after a short amount of time. So this also is some burden on the species. But yeah, people try to develop all sorts of new, less invasive tags. So for example, for some blue sharks, I think, and some rays, they made a kind of harness. So they kind of put this harness made of ropes kind of on the front of the torso of the shark, and then the first pair of fins kind of blocks the harness from kind of disattaching, and then there's a tag floating behind the shark. And because sharks do not move backwards but only forwards, they kind of made sure that this is still attaching. So there are many studies and experiments running for now, but tagging internally is still quite a widely used technique.
And just another thing, because when you tag 30 individuals, then at the end, why do you only recover 11? Or
Yeah, we recovered 8 tags.
because they are lost, or because you don't have the signer, or maybe you said but I miss it.
Well, the tags, they emit acoustic signals.
Yeah.
So if the tags are within the receiver area, and then we go once every 6 months, we go to each receiver to download the detection data. So we don't get the data in real time. And with these data storage tags, we cannot kind of satellite track them because the shark is underneath the surface all the time. So it just records depth and temperature and stores it internally on the tag. And then these sharks either get fished or bycaught, for example, and then fishermen cut them open, and then they find this tag. So this is why the tag also has a bright red color and has like, please return, you get a reward, because it's very important. It's very important data and very, very valuable. So it's really important to return the tags. Some tags also get washed up at the beach if the shark dies and gets decomposed, and then the tag floats up. So the tag floats and then gets washed up at the beach. But it's a lot of money that goes into these tags and little data that comes out. But if we get data, then the data is very rich. So really try to make the most of these 2 year-long depth logs that we got.
Really challenging. Thank you so much.
Yeah. And any questions in the audience? I just have one following on the tags. Like, what are the size of them, and how was the welfare of the animals taken into account for the study?
Yeah, so the tags were 9 centimeters in length and 1.6 centimeters in diameter, which is a fairly big tag. But there's a study that kind of says the tag's weight should not exceed 10% of the body weight of the animal that is tagged. And for us, it was below 2% of body weight, so that's good. But I was talking to some tagging experts in shark tagging, and they said that maybe the tag's volume could have been a bit too voluminous in the shark's body abdominal cavity, because different species have differently loose abdominal cavities. So this is a second factor that should be accounted for, but there is no literature at all on the influence of tag volume on animal welfare. And then for the tagging on board, the sharks, the gills were at all times covered with wet cloth to cool it down, and then the tagging procedure itself was retained under 5 minutes. So from getting the shark out of the water, tagging it, and then releasing it back into the wild, it was 5 minutes.
Mm-hmm. Okay.
Yeah.
Thank you. Well done.
Yeah. Thanks
you.
very much.
Yes, my
If you
memory
pronounce
is.
it wrong, it
No,
doesn't
it's
matter.
okay. No, my memory is terrible.
Okay, so our next speaker in this session is Nargis, who's going to talk to us, also staying in the North Sea, but now on fish communities in the North Sea. So over to you, and good luck.
Hi everyone, welcome. I am Nargis Dukan, and I'm so excited to present to you my master thesis project on spatial analysis of fish communities in the Belgian part of the North Sea using environmental DNA metabarcoding. So let's start. Climate change, overfishing, sand extraction, offshore wind farm installations. Marine habitats face constant challenges related to anthropogenic activities and their impacts. In these conditions, biomonitoring efforts gain utmost importance. Biomonitoring provides us valuable information about status of the communities, effects of stressors, and habitat disturbance. Traditionally, biomonitoring surveys rely on methods such as visual, acoustic, and catch-based surveys. From those surveys, beam trawl method stands out for the Belgian part of the North Sea, since the community composition mostly composed of demersal and bentopelagic fish species. However, as you know, this method is highly destructive, needs taxonomic expertise, and mostly misses rare species and early life stages. All these limitations underscore the necessity of improvement in monitoring strategies. Environmental DNA metabarcoding emerged as a non-invasive, cost-effective, and reliable method that can overcome these limitations and expand the detection spectrum beyond the capabilities of conventional methods. eDNA refers to organismal and extraorganismal genetic material that is suspended in the aquatic environments. And eDNA metabarcoding involves the parallel sequencing of this genetic material for multi-species identification at community level. Besides expanding the taxonomic spectrum, many studies demonstrated the efficacy of eDNA metabarcoding, defining the fish community patterns in horizontal and vertical space. In horizontal space, the method has been proven to be effective for distinguishing the fish community patterns in fine-scale resolution at distances as small as a few meters and also in broader oceanographic scales from tens to thousands of kilometers. And there are still several studies in vertical space, but these studies mostly focused on open ocean waters with pronounced depth profile and also stratified waters with thermocline or halocline. Unfortunately, the areas such as shallow and well-mixed areas, as exemplified in the Belgian part of the North Sea, are very scarce. So, related to that, our primary objective to analyze the fish community patterns in the Belgian part of the North Sea that is characterized with such conditions. And to address these objectives, we answered the following research questions. What is the horizontal fish eDNA pattern in the Belgian part of the North Sea? And does it reflect the community structure obtained from the long-term monitoring data? And what is the vertical fish eDNA pattern of the Belgian part of the North Sea, considering its shallow and well-mixed characteristics? And as a secondary object, we analyze the quantitative power of eDNA, and we address this objective by asking the following question. Does eDNA quantity demonstrate a correlation with actual abundance and biomass of fish species? Before I dive into the methodology, I would love to elaborate a little bit more about our study area. Belgian part of the North Sea is dominated by tide-induced residual currents in the coastal areas and channel currents, channel waters in the offshore areas. And environmental factors such as salinity, temperature, tidal stress, sediment type, and bottom topography shapes the community structure. Ehri et al. In 2009 defined two distinct community structures in the area associated with coastal and offshore waters. In a more recent study, Debaker et al. Increased the resolution of the structure and introduced a transition zone. The community of the transition zone composed of the species primarily occupying coastal and offshore areas, and it exhibits the highest diversity. So, to analyze our special eDNA patterns and quantitative power of the eDNA, we collected surface and bottom samples from 12 stations and bottom samples from 5 stations. And the samples were collected as 5 replicates, and they were immediately filtered on board via a Sterwex filter using a peristaltic pump. And a beam trawl survey also conducted in the same sampling campaign. And the extraction and PCR amplification was performed in ILVO genomics labs, and we used the generated 12S myfish primers to ensure the amplification of both bony and cartilaginous fishes. And sequencing was performed with Illumina MySeq sequencing platform. And after demultiplexing and taxonomic assignment of the amplicon sequencing variants, we analyzed alpha and beta diversity for spatial analysis using 2 factors, depth and zone. And for our quantitative analysis, we compared relative read abundance of the ASVs and with relative abundance of biomass that we obtained from the troll data. And our results. Our study successfully identified 54 species using eDNA metabarcoding, whereas 39 species were morphologically identified using troll data. And in total, 60 species were identified using both methods. From these 60 species, 21 of them were exclusively identified with eDNA, whereas only 6 of them were exclusively identified with troll data. And from these 6 species, Chelidonyctis lucerna and Etriga burnardus has identical 12S sequences. Likewise, Hyperoplus lancellatus and Ovidites tobianus also has identical 12S sequences, which makes them unsuitable for 12S metabarcoding. Except that you see that our false negatives are pretty low. And here we see the bar plots for species richness and Shannon diversity index. X-axis represents the zone factor with coastal, intermediate, and offshore levels. Brown bars represents the morphological data, and dark blue, you don't see the difference, but it's blue and green. Dark bars represents the eDNA bottom samples, and light bars represents the eDNA surface samples. However, our focus on statistical analysis is only on eDNA, so I will only mention about that. So we conducted a two-way ANOVA, and our statistical analysis revealed that there is no significance of the depth factor on alpha diversity indices, whereas zone factor was significant for our alpha diversity analysis. And also post-hoc pairwise analysis showed that the only difference was between coastal and offshore areas. Beta diversity analysis also strengthened the alpha diversity results. We found that only zone factor has a significant effect on our community composition differences, and also pairwise analysis revealed strong results on each level of the zone factor, indicating that coastal, intermediate, and offshore areas has distinct fish assemblages. And in our NMDS plot, you see that NMDS depicted clear clustering for zone factor. Green is coastal, blue is intermediate, and brown is offshore. And you see that there are 2 polygons in each zone, which represents the sampling depth, and they overlap, so it also strengthens our result from data analysis. So to relate it with our objective, we found that discernible horizontal fish eDNA patterns in the Belgian part of the North Sea, and this pattern is strongly aligned with the community structure obtained from the long-term data. And I will give some examples in the species level. We found that Limanda, Solea, and Pleuronectes platessa are widely distributed in the Belgian part of the North Sea. This is highly consistent with the study of Debacker et al., which reports the long-term monitoring, with just one difference, which is Solea is found to be related to coastal waters. This may be attributed to the temporal small-scale variability of aggregating fish species like Solea. And Platychtus phylestus was found to be strongly associated with coastal waters. It also agrees with the long-term monitoring data. And Pomotoschitus species also reported to be widely distributed in the Belgian part of the North Sea. However, morphological data does not provide us a species-level partitioning of the genus due to the challenges about the morphological identification. eDNA metabarcoding elucidates this data by associating Pomotoschitus minutus with coastal waters and Pomotoschitus lawsonii with intermediate and offshore waters. And one of the most interesting findings of our study was Sardina pilchardus. Sardines were found with the highest reed abundance of all species, and they were detected in almost all samples. We can attribute this to the presence of the high amounts of eggs and larvae in the water column, because we didn't catch not even one single individual sardine with the troll data. And another important bentopelagic species, Culupea herengus and Merlangus merlongus, was also associated with the coastal waters. And we found that also with the second part of our first objective, we found that there is no eDNA signal differences in the vertical space within the Belgian part of the North Sea. The fish species that exclusively identified with surface eDNA were demersal and bentopelagic fish species, and they were only detected with very low eDNA abundances and only detected in 1 or 2 samples. Likewise, we found comparable amount of eDNA of pelagic fish species in surface and bottom samples. In that manner, the sampling depth does not reflect the habitat preferences of the species. So these results indicates that the mixing in the area enhanced the vertical eDNA dispersion. And for our quantitative analysis, we selected 6 species depending on their high relative read abundance and the detection at least 10 stations with the troll data. We found a significant correlation between relative read counts and relative abundance and relative biomass for Limanda, Echictis vipera, and Trachurus. For Merlangus, the only significance was between relative read count and relative abundance. For Solea and Chlorenectes pilatessa, we didn't detect any significance at all between relative read count, relative abundance, and relative biomass. So to discuss about the reasons about that, biotic and abiotic factors influencing the eDNA concentration is likely to influence also the quantitative correlation. From those factors, eDNA production rate depends on age, life stage, sex, biomass, and surface area of the organism. eDNA release is higher in a juvenile fish group due to their higher metabolic rate when you compare it with the adult fish group. So if the population is dominated by juveniles and young adults, inflated eDNA signals may influence the quantitative correlation also. Another important factor would be a PCR bias related to primer template binding efficiency. Certain species may exhibit enhanced or reduced amplification due to the variations in the primer binding sites. So to relate with our second objective, we found that eDNA metabarcoding falls short to provide robust quantitative estimates, which is very essential for the fisheries assessment. So for our conclusion, once again, eDNA metabarcoding is proven to be highly effective for monitoring surveys, substantially increasing the taxonomic detection rates. And we also demonstrated the efficacy of eDNA metabarcoding for reconstructing the horizontal fish community patterns that is highly compatible with long-term monitoring, and we did it with just 1 eDNA survey. And also, we found that shallow and well-mixed waters of the Belgian part of the North Sea do not exhibit a vertical eDNA fish community pattern. Our study serves as an example to understand the vertical eDNA dispersal in areas with such conditions. We also found that, unfortunately, eDNA metabarcoding cannot yet replace beam troll surveys for quantitating fish populations. And lastly, further investigation on local eDNA transportation, production, and decay rates, as well as considering the early life stages of the species, will substantially increase our understanding of spatial eDNA patterns and its quantitative relationships. Thank you all for listening.
Thanks a lot. Thank you. So, over to the jury, whoever wants to go first for questions.
So, first of all, congratulations. I really enjoy your talk. So, I think you explained really well, and I think your choice of study area was great, since you have such a nice previous study about the community, the fish community, fish assembly. Yeah. So it's a perfect study system to work with. And also because you have this mixed thing, so that help, I think in some point helps. So well done, well designed. I like it a lot.
Thank you.
I have some methodological questions, and also then other questions. So first of all, of course, you might have certain bias. Yeah, you're using only 1 pair of primers, I guess. You are not using cocktail of primers.
No, only 12 is my fish.
Yeah, that's But in any case, put it like in a positive way, you detect most of the species that you expected to.
Yes.
So in your case, you know more or less what to expect, what to find.
Yes.
So in that case, it was fine. And also, did you find some sort of because that's one thing that I mean, when you go there to a field, even though you have a system where there's a mix, well, I don't know really what the reason, but I trust you that there's a mixed system. Blah, blah. It's the same all across the year. That
well,
mix,
I think
the mixture it's the same all across the year.
I think in summer seasons there is a little bit stratification, but we conducted our sampling campaign in October.
And what about those previous those previous expeditions? That was also in October.
Yeah, the ones I mentioned.
Yeah, the two that one finds two clusters or two assemblies, and the other found 3 assemblies.
Yes,
Were
yes.
exactly the same point.
Yes,
in
the
October.
recent one, the Bakar et al. Was also autumn sampling campaigns,
Okay.
so it was really easy to compare.
Okay,
Yeah.
that's good. Because also it would be nice to have some sort of, I mean, because at the end you go there and you are taking a picture. Yeah, it's of distribution, in this case of molecules, of small molecules.
Yes, definitely.
So it would be nice to see if along different part of the year, if you have the same. And also the other thing that I was thinking, when you analyze that, did you detect some bias in amplification? That means that some species that you expect to be quite abundant that were not there. Did you find some?
Well, I would say for horizontal patterns, we observed some biases when we compared with the long-term monitoring, like Platychtus flessus. In long-term monitoring of De Backer et al., they reported that it's offshore species, but we found that it's in the coastal areas. So we actually, I would say it is about the temporal movement of the species, because the demersal species moves also, not migrates so long, but they moves they follow the food availability, and they move like very small in areas. So that kind of things we observed. For abundance, well, sardine, we didn't detect any sardine, but it was the highest reed abundance in our study. I have to think about that. I have many of them, but from excitement I don't remember now.
Perfect. So that's all. Thank you very much.
Thank you.
And congratulations.
Yeah, thank you.
Well done.
Question from other Germans? Okay, thank you, Lars. Thank you very much for your presentation.
Thank you.
I really enjoyed it. I have a couple of questions. First of all, you conclude that DNA metabarcoding is not good for abundance analysis.
Yes.
But when you perform the Shannon index, you are considering number of reads, right?
Yes, we consider number of reads.
So it's not really informative for them.
Well, we consider the number of species, not the number of reads actually.
Abundance.
Yeah, number of species, yes.
Only
And
number-
also
so what is
for
the difference?
Shannon diversity index, Shannon diversity index takes into account also abundance. What was the question again? Sorry.
Because I think if you can go to the slide, I think you use number of species and also the Shannon index.
Yes, but this is for species abundance. So the correlation we made is for read abundance from the metabarcoding data. So these are the abundance again, but the read abundance that we obtained from metabarcoding. So it's kind of different.
Then when you correlate the presence of the species with more shore or on the waters, is it because you find more samples or because the reads are more abundant?
Well, we find, I also conducted indicator species analysis to understand which species are most associated with which zone. And also we have relative read abundance data. To be honest, indicator species analysis falls short a little bit to really understand all the distribution of the data. I also did simple analysis, but simple analysis also gives you pair-wise comparison, like say Echictis vipera contributes to the differences between coastal and offshore, but it doesn't say which zone this species occurs. So I didn't report that, that's why. And we actually combined indicator species analysis, also relatively abundance, to understand the actual distributions.
And lastly, the last conclusion about all the future investigations or so on. You think EDNA metabarcoding is the best way to do it, or would you consider to use species specific?
For community analysis and biodiversity,
And for DNA transportation as well. And maybe with an approach more focused on species, because you have many different species.
Definitely. I mean, like for eDNA transportation, I mean, if we focus on eDNA transportation a little bit, which we are working on models to track the eDNAs in North Sea. This will elucidate a little bit if there is any disparity between long-term data and the eDNA metabarcoding. And for quantitative analysis, of course, it would be better to use digital PCR or qPCR to analyze it. But we wanted to see if it really correlates, because there are some research that reports there is a correlation, especially for Solea and Pleuronectes pilatessa, with the most commercial fishes. But I think it's a little bit random findings, because sometimes you find correlation, sometimes you don't.
And it depends on the species.
Depends on the species as well, yeah. I mean, like when there is Solea and Pleuronectes pilatessa in the community, they really dominate the PCR, and yeah, it really affects the detection of the other species. Yeah.
Thank you so much.
Yeah.
Yes, yeah, congratulations. I really appreciate it. It's almost the first time I like a presentation with eDNA.
Thank you.
Because maybe I like fish. I can see maybe a perspective in monitoring fish. And it's very surprising how much information you get in those bottles.
Yeah, definitely. I'm always amazed about that.
I spoke to Karim Erzimi yesterday. Someone has been doing the same type of survey in the Bay of Faroe, and consistently they have, the fish that usually fish. It's good, but they have very strange animals. They found a chimpanzee and they found a yak.
Yes.
So why that? Did you find something
I found
very not important here?
I found very interesting things. I found wolf, Canis lupus, and wild boar, chicken, and yeah, human is, I think, in all eDNA method you find it. And there was a fin whale we detected, and it was really nice story actually. There was a stranded whale in Netherlands during the sampling campaign, so we were happy to be able to detect that. But yeah, we discard them for the analysis.
Okay.
So actually, if I can show you.
What does it tell us about the method? Is it why did we find some things which are so strange? If I were a mathematician, I would not admit such mistakes.
I think this is amplification of non-target species is a problem for all primers. 12S is one of the best to analyze fish species, and it detects to the species level. But as you see here, Animalia, Bacteria, Chromista, and we found almost like in similar percentages. So most of the ASVs we detect actually non-target species. So we had like over 6,000 ASVs, and at the end only 200 of them of fish species. So yeah, you have to, in bioinformatics steps, you have to discard them.
Who did these bioinformatics steps? Is it you, or was it provided to you?
Well, the how do you say it? The blueprint, I would say, is provided, but then you have to arrange it according to your own analysis. So we used DADA2 and Metafish pipeline actually, which starts with cutadapt for the demultiplexing and the reorientation of the sequences. Then we go through DADA2 pipeline, which quality clears the less quality sequences, and then also does the taxonomic assignment at the end. But we also like use the BLAST algorithm and NCBI database to.
I can stop you for this question because that is exactly what you decided not to explain, and which make your presentation much better for a non-expert on this technique.
Yeah.
But I have a question also. Okay, you are speaking about vertical mixing, but what about horizontal mixing? There's a lot of horizontal mixing in the ocean.
Definitely. Especially.
So what can you tell us about the horizontal mixing in your place?
Yes.
Your friend?
Yes, that's why I was mentioning about eDNA transportation, because it's a very hydrodynamically active area, and there are very strong currents from channel currents and also in the tidal induced currents. So this obviously affects the distribution of the eDNA, dispersal of the eDNA. But from previous research, we found that there was a research conducted also in Belgian part of the North Sea, comparing the fish communities between offshore wind farms and the outside of the wind farm. So these communities are distinct from each other 5 kilometers or 13 kilometers away. So they were successfully distinguished, these communities. So we could easily say that it's effective in such less kilometers.
How is it possible to maintain offshore coastal gradient if you have horizontal mixing, which is what you observe?
How can I get the question?
How is it possible to observe a gradient in the species community composition between two sites if you have mixing?
Well,
because it's what you are observing. You're observing an offshore community and
coastal.
How can you explain it?
Well, eDNA stays in the, especially in North Sea, maximum 1 or 2 days. So in the normal open ocean, maybe it stays longer, but in North Sea, when the temperatures are around 10 and 15 degrees, it's just 2 days. So we can still talk about community differences in the area.
Okay, but there is no effect on the circulation? Don't you have a kind of frontal structure or the circulation of a current, which is different between the two?
Well, North Sea is a really shallow water, so I think depth is not the main factor there. There is sediment type which is affecting the communities, because the shore is more fine scale sediment, and when you go to offshore, it becomes to be coarse sediment, and this is one of the most important factors that affecting the fish communities, because they're demersal fishes.
One little question. Yes, who were those exclusive species that are found in metabarcoding but not in the trawling and vice versa? Do we have a reason for that? Is it a question of kind of volume which is sampled by
Yes. Well, these species, we can say that they are rare species, but they are indigenous of the area for sure. But we detected them in 1 or 2 samples and with very low abundances, so it's really hard to say if we really detected them or it is false positive. So we don't put our focus on this species. Mostly we are in this intersection areas that covers the majority of the biodiversity. But when so ILVO is a fisheries institute, so they mostly focus on the majority of the things. But if the aim of the biomonitoring effort is a comprehensive effort, then it's maybe better to optimize the sampling effort.
So there's just one comment and question online as well, which I think is a little bit related to something Lars was getting at. So first of all, very clear presentation, well done. And then the question is, do you think the volume of water filtered may explain differences between coastal and offshore samples?
Yeah, definitely, because turbidity is much more higher in the coastal areas, and when you go offshore, you see that the water is getting clear, and we filtered almost 3 times more waters.
Okay.
So it really, I think, affects the detection of the species, and over 70% of our species that are exclusively detected with eDNA samples were in offshore.
Okay.
So yeah, it really
where
affects.
you're getting more water.
Yes, definitely.
Okay, great. Thank you. You've answered everything very clearly. So
Okay.
well done again. And thank
So, final speaker. Perfect. Okay, so we're on to our final speaker in this session. Emiliana is going to talk to us about benthic nematodes and sand extraction. So over to you and good luck.
Thank you very much, Katie. Yeah, we're staying in the Belgian part of North Sea, and unfortunately for you, more eDNA. Good afternoon, esteemed jury and dear guests. My name is Emiliana Tuper-Carabaño, and I'm pleased to be with you here today to present the results of my thesis, eDNA metabarcoding of benthic nematodes and machine learning for sand extraction impact assessment. And I know that's very wordy, so we'll dive right in. The sand extraction industry began in Belgium in the 1970s, and it's been increasing steadily ever since. And it is primarily destined for coastal protection as well as construction, such as in asphalt and cement production. And we are bound by laws such as the Water Framework Directive to monitor the states of European waters and strive to have good ecosystem health. And one of the ways in which we do this is through biotic indices, where we calculate the state of an ecosystem deducing from the species that are present, their sensitivity, tolerance, abundance, etc. This is what the suction dredger looks like as it dredges along the sea floor, and you can imagine that it has a lot of effects on the benthic communities. And specifically, the biotic index that we use in the Belgian part of the North Sea is BECI, or the Benthic Ecosystem Quality Index, and it is based on macrobenthos, which are organisms larger than 1 millimeter that inhabit the seafloor, like sea stars. And it relies on taxonomic experts that manually sort through the samples and identify them, which, as you could imagine, has a lot of challenges. For example, it is very time and labor intensive, which can lead to it being expensive. There's disagreement between experts, and there's a dwindling number of people that are specializing in this expertise, which can pose an issue in the future. And so this is where new technologies can come in to circumvent these challenges, namely environmental DNA or eDNA metabarcoding, as well as high-throughput sequencing. And with these techniques, not only work in water, but also we can take sediment samples from which we can extract, sequence, and compare it against the reference database of the DNA to see which organisms are present. And while these technologies have worked to successfully calculate biotic index, they suffer when reference databases are not complete, which is often the case with smaller organisms like meiofauna or bacteria. And so this is where our second tool in the toolbox can come in, namely machine learning. And so we can train an algorithm to predict ecological status by feeding it the ecological status or the macrobenthos information we know alongside eDNA data. And once the model is trained, it can predict the ecological status. And why is this important? Why should we care? Well, one of the main advantages is decreasing the cost, and we know how important this is in science and life as well. And this would lead us to have a higher spatial resolution as we can sample more and ultimately have a greater idea of what's truly going on in the ecosystem so that we can sustainably exploit these resources while still benefiting from the ecosystem services that these organisms provide. And this is where our study comes in, because it has been shown that area and disturbance-specific protocols are required. This cannot be applied universally as areas differ and organisms present differ as well. And this project is part of a larger project at ILO, where we're exploring these techniques for different benthic groups using different gene markers. In this case in particular, we use the 18S gene marker to target Nematoda, which are the most dominant taxa of meiofauna. Meiofauna are organisms between 45 micrometers and 1 millimeter that also inhabit the seafloor. These are less typically used in monitoring because they are a lot harder to taxonomically identify. But we can use eDNA to solve this problem. So specifically, what we decided to look at and explore was 1,
can we detect the effect of sand extraction using nematode eDNA metabarcoding data? And if so, can we use this data to accurately predict the ecological status using Becky? Concerning these issues of taxon-free, taxon references, and databases, we also decided to add a small tertiary question about if the taxon specificity of our eDNA metabarcoding data affects the results of our previous two questions. So, onto our methodology. We're keeping in the Belgian part of the North Sea, and the monitoring campaigns yearly sample 3 different sandbanks where sand extraction takes place, namely Thornton Bank, North Hinderbank, and Postic. So we were lucky enough to have data for 2019 and 2021, which allowed us to look at interannual variability. And having 3 different sandbanks allows us to look at interbank variability. And this is very important because these banks have different local geologies, which of course affects the species and the community composition in each bank. They have been shown to be different. We had a total of 86 samples, and we classified them to sand extraction impact level according to the volume extracted in the year prior. And so we had low, medium, and high, and we also had reference samples that were taken from areas within the same bank, but where no extraction activity takes place. And for a quick scheme of our project, the sediment sample collection consisted of simple Van Veen grabs, and from those we took 20 millimeters syringe to extract DNA, and we sequenced it, and then we used also the data2 pipeline to obtain our ASVs, which are ampli-consequent variants. And for those not in the know, ASVs basically refers to the unique sequences of DNA that we can retrieve from our high throughput analysis. And once we assigned taxonomy to them of nematoda, we only took the nematoda and we did a diversity analysis. Alpha diversity, meaning the specific diversity in each site, and beta diversity, which looks more at relative to different sites. So to answer our second question, can we use this data to predict ecological status? We continued with the traditional morphological assessments. We had great experts at ILVO that manually sorted our samples. And when we had the macrobenthos parameters that we used to calculate Becky, we fed these parameters alongside our DNA sequences into an algorithm, namely the least absolute shrinkage and selection operator, or easily said, lasso regression. And we chose this model because it can handle high levels of co-linearity between predictors, and it can also handle having a larger number of predictor variables than of samples, which was our case. So once the model was trained, we calculated the Becky index twice. Once from the normal method using macrobenthos, and the second time from the predictions of macrobenthos parameters that we derived from the model. And finally, we compared the 2 assessments to see the level of agreement between the 2, basically seeing if the predictions are accurate. Lastly, for our third question about taxon specificity, we repeated this process twice. First, on all the ASVs classified to nematoda phylum, which we named ASV level, and then to all the ASVs classified all the way down to genus level. So for instance, in this case, both ASVs will belong to the ASV level dataset, but only the second one would fall under the genus level dataset. All right, so on to our results. This is a graph of our different diversity metrics, namely richness, Shannon-Wiener, and IMBRSea. We have our impact levels in the x-axis and our different sandbanks by the boxes. Right off the bat, you can see that there is not a lot of differences, and we see a lot of overlap between our box plots. And this is confirmed when we run our ANOVAs, that basically there are no significant differences in sand extraction impact level, nor bank, and not even year. And for genus level data, not shown here, we do see some small differences regarding Shannon-Wiener and IMBRSea. So how is this related to the literature? Previous studies that have looked at long-term effects of sand extraction impact are actually consistent with this information, especially the ASV, that long-term extraction impact, we don't see a difference in alpha diversity metrics. However, the community composition, meaning the organisms that make up that diversity, do indeed change according to sand extraction. So we're seeing a change in the community, but we remain with the same numbers of organisms present, basically. And this is indeed what we see when we look at our beta diversity. Here we have an MDMS plots of all the sandbank combines. And this should be blue, this should be purple. We can see that the sandbanks, the sites are clustering according to sandbank. and not very much according to extraction impact level. But if we look at Thornton Bank specifically, there's two glaring things in our chart, and mainly we see a significant separation between years, so we see a lot of yearly variability, but we also see a clustering of our impact levels, with the blues being medium and high extraction impact levels clustering in the upper left, and then the greens being the low or no extraction impact levels clustering mainly along this diagonal in the lower right corner. And this is indeed confirmed when we run our Kramer-Novas, and our post-hoc test indicate exactly this, medium and high versus low and reference. And while we do not see this pattern as clearly with genus level data, the results of the statistical analysis reveal the same. And so this is consistent with the studies from Barnaver-Becker et al. That community changes while alpha diversity stay the same. All right, so basically to sum that up, we can see the effects of sand extraction on nematodes using this data to some extent, namely in Thornton Bank. And it turns out that Thornton Bank is the bank that experiences the highest intensity and the highest frequency of extraction, while the others exhibit either low intensity or low extraction volume. And comparing with the literature of what has been found in the field before, we see that ASV level data is more consistent than genus level data. And one of the reasons for this could be, as I showed you before, when we go all the way down to more specific taxonomic assignment, we lose a lot of resolution in our data. So by using ASV level data assigned to nematoda, we can make sure that we're targeting the species that we want without getting monkey or humans, but also without losing as much resolution if we try to go to specific organisms. So, given this result, we would expect that the model then works really well and we get great predictions. Not quite so, unfortunately. Moving on to our machine learning part of our model, of our project. Here is a comparison of our assessment from true macrobenthos and from our predictions. And basically, it's a deviation of the predictions against the macrobenthos assessment. If we have a 0 on the x-axis, it means that we have an agreement between the two. If we have positive, it means that we're overestimating, like predicting a good from a true moderate, and a negative is an underestimation, so predicting a poor from a true moderate. And so right off the bat, if you look at the percentage of correct assessments, meaning the zeros, we can see that they're not that great. For example, we wouldn't want to be right just 60% of the time, let alone 40% of the time, at least not me. And this is indeed confirmed when we run our statistical analysis, namely our Goins kappa coefficient of agreement between two raters. So if you look at our alpha level, RSV level, we see that we have no significant agreement between the two. And if we look at our genus level data, we do get a significant fair agreement, would be qualified, but this is not high enough. We would want a kappa coefficient of at least 0.8 to be able to rely and trust our prediction, which we don't have. So to answer our second question, no, we can't really use the DNA metabolic coding data of nematodes to make predictions. And one of the reasons that we think that might be behind this is because the studies that have used these techniques, namely using a different taxonomic group for a macrobenthos-based index, have been based on chemical disturbances. For example, oil spills or organic enrichment. Whereas here we have at hand a physical disturbance that is sand extraction. And so we decided to look up more into experiments or observations that dealt with macrobenthos versus nematoda or macrobenthos versus meiobenthos. And we see that for chemical disturbances, they're both affected by the disturbance in a way that is correlated, and thus predictions turn out to be more accurate. But when we look at physical disturbances such as sediment deposition, we see that both groups are affected, but they're affected in a way that is not correlated. And this could explain why for our model, mainly the lasso regression, we don't have great predictions, because the best predictor variables in a lasso regression are those that have the highest correlation with our dependent variable. And so we looked at studies also that didn't quite look at sand extraction impact, but they looked at indirect effect of sand extraction. For example, sediment size change. The more sand we extract, the deeper we go into the geological layer, which have different sediment sizes. And they have found, this was indeed the case for Barnaver-Becker et al. In 2011, that they found that they're both affected, but while nematodes exhibit a drastic change in community after a certain level, macrobenthos seem to be a more gradual change. And one of the main takeaways from this is that we have the importance of developing area-specific protocols, because what has worked for other places has not worked here. For future studies, we would love to have more data, which is a limiting factor here, of different banks, because we know that they're different, and we could perhaps see more higher resolution with this. And of course, use a COI gene marker targeting macrobenthos, as they're more likely to be correlated with the true macrobenthos. Other features that worked well was filtering the number of reads by the number of ASPs by presence or prevalence in more samples, and also applying abundance classes as opposed to raw read number to avoid biases to organisms that are amplified or get detected better. So if there's two things that you should take away from this project, is one, that eDNA metabarcoding can be successfully used to assess diversity patterns in these communities. And this in and of itself is pretty innovative. Using 1 gram of sediment and a lot less labor intensity, we are able to see the same patterns that we can look at taxonomically. And this in and of itself, first time in the area, first time to the type of disturbance, is an achievement. But there's another big important factor that our data is telling us, and it's that these macrobenthos-based indices are not giving us the full picture of what's going on in our ecosystem, since we see that there's an effect on nematodes that is not reflected on this index. And so with this, we join a number of authors that encourage the incorporation of more benthic groups into our biotic indices. And while I might be a little biased, I think genetics is the way to go with this, but it has also been shown to work, as we can indeed detect and differentiate between species that are too hard to detect by eye, such as meiofauna and bacteria. And with that, I would like to thank you very much for your time, as well as all the data sets at the EVO, all the hands that help prepare these data sets at the Marine EVO Lab. Thank you very much.
Well done, Emiliana. Thanks. So, Jury, you want to kick off this one?
Yes, thank you. And thank you so much for your presentation. I think it was great. It was really clear. So I have only a couple of questions. In the quality indexes, do you know if you are overestimating or underestimating the status or the in most of the cases?
Yeah, actually, if you look at the...
For me, sorry.
No, you're good. If you look at the graph, we actually thought about that, and it's more or less evenly distributed, I would say. We see that it might be a little bit, but it's not a glaring difference. It's not, we're not very heavily skewed to either side or another. And if you were to ask me for the reason for this, I think it's because the banks behave differently. So when we look at the banks individually, we would see that it would underestimate for some and underestimate for others, which turned out to balance once you combine all the data.
I think maybe it has something to do as well with the references of nematode in the databases.
So for the reference databases, we actually started with a very specific reference database, because it's been shown that the smaller your reference database, the less competition there is in like the classification, using especially the RPD algorithm that we used. So we were able to assign a really large number. We had 16,000 ASVs to nematoda. So I don't really think it's because of the classification, which could be, but I really do think it's because even if we had a great classification, they're still behaving differently to the macrobenthos. So no matter how specific we were, I don't think we could ever get the best prediction, since they don't really correlate to each other.
Okay. So if you use the same traditional method, maybe you find the same than with environmental DNA?
And I think that's why I think the suggestion of using COI gene marker for macrobenthos would be the best idea to go from now on, because they would most likely be correlated, at least more than nematodes. And this is actually happening. My supervisor is part of her PhD project, so she's looking at COI for macrobenthos and 16S for bacteria.
Okay.
But based on this, I think probably macrobenthos using COI would probably be the right choice.
Okay. I think that's it for me, Brad. Thank you so much.
Thank you.
Yes, I prefer with the fish, but the story with the sun was also very beautiful. So congratulations.
Thank you
for it. So yeah, what is the part of your job into it? Did you count the nematodes?
Oh, thankfully no, because that's a really hard job. People at the lab were saying that indeed they spend so many days with one single sample. But no, so I came in and the DNA was already extracted. So I did the rest of the PCR protocol. We had amplicon PCR, index PCR. And then once we send the samples for sequencing, we get them back demultiplexed. And from there we run cleaning them, trimming them, then verification as in taxonomy, and then all the statistical and diversity analysis.
Going back to this figure, I'm surprised that the accuracy, I think we can speak about the accuracy of detecting the community composition here. Why is it so low? For me, we, as you know, we do a lot of machine learning on plankton community based on images. I think sometimes our accuracy are kind of better than that. 56% is low.
Yeah, no, definitely. I think that's why. So it's the accuracy of our model, like the accuracy of using nematodes for predicting macroventas.
Yeah.
I think that's why the accuracy, the relationship is not there. We ran a posterior correlation analysis, and we do see a very low correlation between nematodes and macroventas. Like we might see one ASV that we don't know who it belongs to that is super correlated, like 0.8. But it's very little if you have 14,000 ASVs. We don't really see it reflected in
the ASV are less.
That's a very curious part, right? That we see ASVs are better for the diversity analysis, but it turns out for the model, it's not as good as genus. And I've been pondering on that question for a while, and I've been thinking, well, maybe the genus that gets detected, for example, are larger ones or ones that are more studied, that might be more studied because they're easier, and so maybe they relate more to macromenthus because they're bigger. But I think there's a myriad of reasons why that could happen. But at the end of the day, since none of these are good enough to detect,
it's not a question of the level of recognition, because ASV there can be a lot of confusions. One ASV could contain different species, while in the genus there are less species in the genus.
Well, yeah, this is exactly. You would have,
and therefore you have more confusion in your results.
Coming in?
I don't know. Maybe I need to think about it, but I was surprised by the.
Yeah, no. If you have an answer, let me know, because we've been thinking about it a lot too, and we still don't know.
Okay. Then maybe another question is, yeah, I did not also understood the whole story about the relationship between the age since the bridging and the change in the community composition, but not the change in the diversity.
Sure.
Yeah.
So do you mean the change in community composition, but not in alpha diversity metrics?
Yeah,
exactly.
Sure.
So
just because we have other species, but with the same proportion each time, so the channel is not changing.
Yes, exactly. So what we found from studies looking at more specifically sediment granulometry, so not sand extraction itself, but an indirect effect of sand extraction, is that as the median size of sand grains change, the communities of nematodes change. For example, when you have very fine sands, we see more filter-feeding, non-selective nematodes. But when we have coarser sands, we see larger predatory nematodes. And so we see that there's a spectrum of the communities that change according to the sand of the sand size. But we always have nematodes present. It's just different nematodes that respond to probably more the biogeochemistry that's happening in the sand, depending on the size and depending on the permeability and all of that. But we always see nematodes.
Yeah, I think this concept is probably well known, that the diversity might stay constant, but the community composition and then the functionality also might change.
Mm-hmm.
So the message that maybe we do not hear enough, not among the scientists, but in the media. not all diversity are
It's
at
the same.
impact.
Absolutely. Yes, they were talking about
And
today.
it's a clear example, I think.
Yeah, and that's why I think we should integrate it into the models because they would definitely change what the ecosystem services or what's happening there, but we're not considering that. But there was a talk earlier today about the wind farms and that they see an increase in diversity, but what is this good if this has never been this way? Like, are we manipulating? Like, it's not necessarily good, right? So you're absolutely right.
Okay, so first of all, congratulations. I really enjoyed your talk. I think
Thank
you
you.
presented it in such a clear way. Also, the way that you use your slides was really, really nice. So congratulations. I think it's a nice study. Also, I think that you, as in the previous case, you are in a really lucky place to study. Yeah, it's a well-known system because even in a really well-known system, you have certain certain difficulties, yeah? Imagine when you try to implement it in a region where it's not well known, the database is fully incomplete, and so on. So in this case, it's fine. So I also would like to highlight it's more like a common, what you just discussed about the number of species and the type of organism and how that changes. So I guess, what's your recommendation in terms of, because for example, when you are analyzing impact of certain region, no? There's like some flag species that if you detect that flag species that has certain, weight in the way that is the impact of that region. So which will be based on your result, which will be your flag species to determine huge change in the ecosystem or in the community, let's say?
Sure, that's a good question. If we had more time, I would have loved to have done a simpler analysis to look at which of the species or the ASVs or which genus are driving these differences. And it's a kind of hard question to answer because some banks see a coarsening of sand sizes and some see a fining. So I think it would be hard to choose one, but I'll try to choose representatives of predatory nematodes like Anoploides, or I use representatives of finer filter feeders. I think Romalorita would be an example of that, I'm not sure. So I would try to look for the representatives of finer and coarser to try to gauge the full spectrum. But indeed, it's interesting because with these analyses, they're good for detecting the patterns, the diversity patterns. But of course, if you want to look at exactly the diversity, it's not as high resolution as if you do a bulk analysis, like if you take all the samples and because we only use 1 gram of sediment, which is amazing because it's only 1 gram, but if you want to look at exactly who's there, you probably want to take a lot more. So it's good for overall patterns, but if you want to look at more into specify which species and the number of species, I think bulk DNA, where you take all the organisms and you make them into a nasty little mix, and then extract DNA from there, you do get more high resolution.
Okay, another thing is more like a methodological thing. Of course, here you also bias. I'm really focused all the time with bias, but PCR bias, it's really contributes to some noise, or at least to some avoid some detection of certain things. So, and related to that also, how ASB could be also affected by that. So, because certain variants you will detect will get amplified, but not others. And that's depending on the genus, that could change completely what you are seeing. So, which kind of solution would you propose? Which is the effect of this, for example, sequencing error in ASB? What do you know?
Sure, that's honestly one of the reasons why let's look at nematoda as opposed to look at anything that 18S primers can detect, because we do see a lot of PCR errors that we would inflate the diversity that we have. And then by looking only at nematoda, we are hoping to reduce so that we only get ASVs that get assigned, so not all the random ASVs that can be produced. But then, of course, there's a lot that went into the model, but one of the things we did to try and reduce this PCR bias was to include abundance classes instead of full sequence read. So we only assign from 4 to 5, depending on the percentage of reads, so if we have one organism that gets very picked up and very well amplified, it doesn't skew the data, but if it's this difference from 5 to 0 versus a difference from 500 reads versus 1,
we try to minimize it that way, and it really worked out for us because when we looked at the predictive accuracy, it was better when we used abundance classes versus when we used raw sequence read. So that's the plus side of using taxon specificity, that you kind of reduce a little bit of that bias.
And what about, do you have, because in your case, it's nice to say, oh, out of 1 gram, yeah, it's nice, so it's nice. But also in your case, it's not a big deal, no? Since, I mean, you could take much more, it's not a limitation, so maybe even some PCR-free. I know that nowadays it's not available, but in the future it could be. Also, I mean, in your case, it's not so difficult to get it, so to reduce some bias, reduce some bias.
To filter more samples, you have more higher amount of.
I mean,
yeah,
I think everything.
absolutely.
I mean, even in genomics, everything is moving towards PCR-free methods. I mean, just to get rid of at least part of the sequencing error. So that's a thing. The other thing is, did you do some replicate, some control condition that you could say like, okay, I'm really trusting my data, you know? Can you comment on that?
Absolutely. So for with actually the 1 gram. For each, we had 3 technical replicates. So from 1 Venvin graph, it was 250 micrograms that we extracted CYAN from 3 times, and then we pulled that, and then that's where the 1 gram is actually 750 micrograms. And for more than the PCR, what we did in the model to really make it more accurate was we had a lot of, we did 10-fold cross-validation. So we would really mix up our training sets and our validating sets, and we would do this 10-fold cross-validation, and we did this 100 times. So the processing time was a lot longer, but it allowed us to, 86 samples is not a lot. Like if you look at a lot of machine learning models, they have thousands or hundreds of thousands of samples. So with this, we try to not get them so that the model is not overfitted to our data. And that's one of the ways. So we did 3 technical replicates and then a lot of cross-validation.
Thank you very much. Well done.
Yeah, thank you.
Thanks a lot. There, if you don't mind 1 more minute, there's a quick question online as well. So the comment is fantastic presentation, and then the question is generating new indicators using DNA is a very hot topic. Where should we start, and are there already DNA-based indicators available?
That's a good question. It's really interesting because indexes are different. This one, for example, works on its relative. So it looks at comparing the samples that we have against the reference data sets, and that's why we have references. And these are a little bit harder to develop protocols. But we do, for example, in IMBRSea, which is one of the chemical disturbance indexes that was developed here in Spain, actually, it's more based on the species present and their level of sensitivity. So if you have one particular species and it's very sensitive, that goes into calculating the index. And for those ones, they have tried to develop, they call it the G-AMBI, which is the genetic AMBI, and for that one, they have been simply replacing the identification of taxonomy from manual to genetic, and for that one, it has been shown to work. They do have the issues of abundance that we mentioned in the previous talk, so they look at mostly just taxonomy instead of abundance, but I think there could be a way, it's been shown to work to that extent, but for these, we need to do a little bit more work.
Okay, thanks very much.
Yeah, thank you.
Okay, so that finishes today's session. So just a last round of applause for all 3 speakers. Well done. And the very best of luck to the speakers who are waiting for tomorrow. We start again in the morning at 9 here, so we'll see you for 2 sessions tomorrow morning again. And this evening, I think there's an activity, you're due to get a message in the chat, I think, so keep an eye on that. And there's an activity at the residence later, so enjoy that. And see you tomorrow.
Thank you.
Thanks. Do you want to turn off this?
Yeah, stop streaming.
Yeah. Yeah.